And we have a machine that the orbiter of mass spectrometry. Though the people who are working in the mass spectrometer, you know that orbiter of mass spectrometer is this powerful mass spec. And with this mass spec, we can identify large number of proteins in the blood samples, in the plasma samples. So this work is initiated by my PhD student Apavito and postdoc Komalika. Apavito is now in America. So meanwhile, we do the proteomics. Before we do the proteomics, we also try to utilize this sample, the blood samples, because this is a huge the resource. So we try to find out some of the markers which are designed based on a hypothesis. Since at the time of the heart attack, the platelet count is increased, platelet become more active. So there is a possibility of releasing some proteins called STLT1 that we hypothesized from the literature and our other works. So we found that STLT1 is the protein which is released from the blood at the time of the heart attack. This is the patient sample, and you can see there is a huge amount increase, several-fold increase in the STLT1. This is a soluble TLT1. This is increased in the patient, ACS patient. ACS means heart attack patient, and more. It goes down not very to some extent when the coronary artery angioplasty is done. So we have collected samples before coronary angioplasty and after coronary angioplasty. And also we have a controlled samples. But at the same time, we have some interesting group, that is asymptomatic groups. Asymptomatic groups are the people, they are not diagnosed with coronary artery disease. They are not going to the hospital for with chest pain. They are normal. They are actually having normal life, but they have some risk factors for atherosclerosis, like high blood pressure. They have unmanaged cholesterol. They may have, some of them might be having the medications for managing cholesterol. So they are called asymptomatic groups. So very interestingly, this protein is significantly increased in the asymptomatic groups, but they don't know there is a heart problem, and which is very high in the patient samples. So that makes it suitable for studying further for making a potential biomarker. So that's what we are working now in further detail to find out whether this can be utilized as a biomarker in large number of patients. So with many other experiments, we have shown that this protein STLT1 binds to the Fc gamma receptor 1, and it activates the inflammatory signal inside the macrophage. We have utilized macrophage culture. I will show detail in other experiments. But for this one, I have just summarized that this protein STLT1 we have shown first time that is the inflammatory marker. It induces inflammation in the system. It helps the cell to maintain the inflammatory stage, which is not good for the system, and then TNF alpha is secreted, and the inflammation stage is maintained. So we have published this paper in Clinical Science. This is published as a cover page article in 2019 November issue. So now I come to the case control design of the human study, the proteomic study. So we have say 9 control and 9 STEMI cases, the MI cases, and then we pulled separately, and we have subjected to arbitrary mass spec at the same time SWOT analysis. SWOT is a powerful mass spec which can be utilized for the quantitation, and arbitrary is utilized for the relative quantification. So we have conducted the mass spec and analyzed the data with the computational methods and validated by wet lab experiment. So these are the data we collect from the human sample, because we need to know the demographic parameters so that we can clinically correlate several the proteins which are altered in the blood sample in later phase. So you can see that we have our powerful arbitrary mass spec could identify more than 2,500 proteins in the control and also in this MI patient. And these proteins are divided into 423 proteins are actually common in both the groups, and 264 proteins are given the control. So these are further analyzed using the sieves of sieve for the volcanoplot analysis, so that you can see the distribution and the p-value of the proteins. So with this sieve software, we can identify the differential express proteins. This is basically a relative quantification based on the algorithm, and we have found that these are the proteins which are altered in the blood samples and the ACS samples. So these are the proteins like PGL-RP2, ABCA5, AZGP1. Some of the proteins are very interesting. We have subsequently validated and conducted detailed experiments. So we, at the same time, the blood samples were also subjected to SWAT MSMS analysis. This was done in NCL Pune in our CSR laboratory in the Mahesh Kulkarni's lab. So Mahesh has nicely shown these 64 proteins are identified in the SWAT MSMS, and out of the 26 proteins are downregulated and 38 proteins are upregulated. SWAT is the sequential window acquisition of all theoretical mass spectra. So it's a very powerful tool. And then we've divided the proteins which are found in the orbit trap and also found in the SWAT MSMS. So we decided that C17ORF1757 is the annual ABCA5, one is downregulated and is upregulated for further study. Similarly, the proteins identified with SWAT MSMS like calcine, PGL-RP2, and AZGP1, these are the proteins we selected for further study. Why do you can ask the question what is the rationale for deciding this protein for further study? Basically, there are two reasons. Number one, these are the proteins which are actually based on pathoanalysis. I am not showing this here. Pathoanalysis found to be related, involved in the cardiovascular disease, the pathophysiological process, and also these are the proteins that we can find antibody reagents for the validation. But there are many proteins which could be validated further. But because with our insight, we decided, okay, these are the proteins to be validated. So we have performed signalome analysis, and based on that, we found that and also IP analysis. IP is a very powerful tool software for analyzing the pathways, and we have found that these proteins belong to transporters and inflammatory and coagulatory proteins. So basically, this is as per our hypothesis that we expect that many proteins will be involved in the inflammatory pathway, and those are altered here. The 6 previously unreported proteins were chosen for further biochemical validation. And these 6 proteins are either related to RCT, reverse cholesterol transport, or development of plaque morphology. So that's the reason we selected these 6 proteins. What are these 6 proteins? ABCA5, C717ORF17, C17ORF57 is downregulated in our orbital mass spec and SWAT MS. And here also we have found that this protein is downregulated by Western blot analysis. Similarly, ABCA5 was found to be upregulated in our mass spec. Here also we validated and we confirmed that ABCA5 is upregulated. So, and these proteins are also checked before and after PDCA. PDCA means coronary angioplasty. Before coronary angioplasty, this protein was increased, and after coronary angioplasty, this protein is decreased. Similarly, AZGP1 is a novel cytokine, PPAR gamma, PGL-RP2, calcine. These proteins behave similarly as we found in the orbital mass spec. So then we selected AZGP1 for further study. AZGP1 was found to be novel adipokine, decreased in the STEMI. It was found to be connected with most of the candidate proteins, so it was selected for further downstream study at the cellular level. We using the in silico analysis using class Pro 2.2, PathDoc, ADOC, we have shown that this AZGP1, this novel the adipokine, binds with CD36. And CD36 is a protein which resides in the macrophage cell surface. So it binds to macrophage, and then what does it do? So does it do? We use monocytes, activated monocytes into macrophage, convert monocytes into macrophage, and macrophage is activated by using oxidized LDL, and macrophage is transformed within 24 hours. It is transformed into foam cells. So you utilize this in vitro system. We have developed this in vitro system. So THV1 is the human macrophage, monocytes. Monocytes is transformed into macrophage, and macrophage is transformed into foam cell by oxidized LDL. And then we found that CD36 is upregulated, and expression of PPAR gamma is downregulated in this system, which is found in the human blood sample. And then with several other experiments, I published this paper in Journal of Proteomics recently, last year. We have shown that the AZGP1 is a novel cytokine released from the adipocytes. So AZGP1 binds to CD36 and helps the conversion of foam cells. So macrophage is converted into foam cells. But this is not an issue. it remains in the balanced stage as long as ABCA5 is upregulated. But when ABCA5 is downregulated, when ABCA5 is upregulated, the RCT imbalance happens and PPR gamma is downregulated. So this process is driven towards the inflammatory stage. So when other molecules are in the normal state like HBCR2, C17 or F7, 57, PPR gamma, this is balanced. This remains balanced. The foam cells, macrophage circuit is balanced. So HDL helps removing excess cholesterol from the blood to the liver, and system remains balanced. But when PPR gamma is downregulated, the foam cells converted into the inflamed foam cells because of the induction of chronic inflammation. At that time, PGLRP2 is upregulated, which is found in the blood of heart attack samples, is upregulated, and then it remains in the inflammatory stage, which is not good. It actually drives the system towards the advanced plaque and eventually plaque ruptures. So that partially explains how the atherosclerosis becomes the transform into inflammatory stage and going towards the rupture stage. So now let me check the second part. So what happens to ABCA5? So we validated one protein that is ZGBC1, the double side adipokine. Now I switched to another protein, which is ABCA5. ABCA5 was found to be upregulated. That was in the blood samples of human patient. So the question is why ABCA5 is upregulated. To answer this question, we have utilized three systems. One is in vitro culture system, cell, another is animal model, and the animal model is a genetically modified animal model. The two animal models, apoe knockout mice, which is very popular for atherosclerotic research, and there is PPR alpha knockout mice, which is endogenously because of PPR alpha knockdown, the cholesterol level remains very high in that mice, and also the human sample. So let me show that the study design. So Olipto is a graduate student. He developed this system. He actually tried to check the data in the in vitro system. So what is happening in the in vitro system? So he utilized he developed the loading and unloading system in the macrophage. He cultured the macrophage, and then macrophage was treated with cholesterol. And he has checked whether what is happening to in ABCA5. So let me tell you, I forgot to mention that ABCA1 is a primary transporter, glucose transporter in our system. That is already known. But nobody has shown that ABCA5 is altered there. So first time we have checked, we have found that ABCA5 is upregulated in blood. So now we are checking in the in vitro system. So in vitro system, he has loaded the cells with cholesterol, and with increasing duration of cholesterol loading, the ABCA1 RNA and ABCA5 mRNA are increased. And also they are removed. When HDL was utilized to remove the excess cholesterol, the ABCA1 and ABCA5 levels downregulated. So it suggests that ABCA1 and ABCA5 behaves similarly as a cholesterol responsive gene. So you treat the cell with a high cholesterol, ABCA1 is increased. You treat the cell with high cholesterol, ABCA5 is increased. But this cholesterol level is the physiological level. Now he has loaded the cells with higher than physiological level. 100 micromolar cholesterol is the physiological concentration for in vitro study. He has increased the cell cholesterol loading with up to 150 micromolar. So that is very interesting. Now you can see that after 150 micromolar cholesterol treatment to the cells, the ABCA1 is downregulated. And at the same time, ABCA5 is upregulated. So this is actually the replication of the MI patient, the myocardial infarction patient, heart attack patient. The cholesterol ABC1 level was downregulated and ABCA5 was upregulated, found in our orbitrap mass spec analysis. So that we reproduce in the in vitro system. So he has developed the system where we can load and unload the cells and show that if we increase the cells with a high concentration of cholesterol, the ABC1 is downregulated, and if we increase the cells with high cholesterol, at the same time, ABCA5 is upregulated. So with this, we again checked in the by mass spec, so validated whether really it is ABCA5. Yes, he has treated the cells with high cholesterol concentration, and then he has shown that ABCA5 is upregulated in response to 100 micromolar and 105 micromolar cholesterol. So not only Western blotting analysis, in fact, in future, the most of the data will be validated by Orbitrap, or mass spec. Not only Orbitrap, any mass spec, because Western blot gives a lot of artifact. So this is a validation by mass spec that yes, it is ABCA5. And then this induces the several other pathway related to atherosclerosis. Now, what is the molecular mechanism? What is the effect of cholesterol overloading at the molecular level? What is happening in the molecular level, transcription factors level? Sorry. LXR1, LXR beta, PPR gamma are the transcription factors regulated, which these transcription factors regulate the transcription of ABCA1 and ABCA5. So, with increasing concentration of cholesterol, LXR alpha is upregulated, but at 150 micromolar, LXR alpha is downregulated. Similarly, LXR beta and PPR gamma. So you can see that these are the transcription factors downregulated. So it could be the reason for why, it could be a reason for lowering the ABCA1 concentration in the blood. That was in the in vitro and also in vivo. And that was also same for the duration of increased duration of time. So if you treat the cells for chronic period like 72 hours, the LXR alpha is decreased. That is why the cholesterol ABCA1 level might have been decreased. And what is happening in the cell? So in the cell fat, you can see this is very interesting data. The cells, as long as ABCA1 is there, ABCA5 is also there to certain level. The cholesterol level up to 150 micromolar, the cells going to the necrotic cells are reducing in number, but the apoptotic cells are going increasing in number. So cells are going towards apoptosis, but mostly early apoptosis, some of the later apoptosis also. So cholesterol overloading represses transcription factors regulating lipid metabolism and increases the apoptotic phenotype. And so we checked using the immunocytochemistry whether these transporter they behave like transporter. So immunocytochemistry study of raw 264 cells under different cholesterol doses to find the localization of ABC1 and ABC5. So ABC1 and ABC5 they co-localize in the cell surface in the raw macrophage cells, raw cells, the macrophage cells. So, and now we checked whether ABC1 can be increased with this LXR agonist. So we checked by the small molecule activator agonist. When LXR activity is increased, ABCA1 is increased. But when LXR activity is increased with the help of GW3965 in the presence of cholesterol, when there is a high cholesterol, ABCA1 is decreased. But at the same time, same condition, ABCA5 is increased. So wherever the situation is there, ABCA1 is decreased, ABCA5 is increased. So it looks like here we got the clue that ABCA1 and ABCA5 concentration expression are like compensatory. They are behaving compensatory process. So that is why in a condition when ABCA1 is decreased, ABCA5 increased. So that's give an interesting clue, and that was produced in with the different kinds of agonist and antagonist. And also we have checked that whether lipid modulators, what is happening in response to lipid modulator like atorvastatin. Atorvastatin is a statin which you know is used to inhibit the cholesterol synthesis. So if you decrease the cholesterol by atorvastatin, ABCA1 is decreased. But if you increase with a high cholesterol concentration, ABCA1 is certainly increased to some extent to manage the cholesterol. But ABCA5 level is increased very significantly because ABCA5 is the cholesterol. ABCA5 is a transporter which could be the sensor for high cholesterol load. So that I am trying to drive you towards that direction, and which is as true in the presence of oxidized Ldl and other stimulator. So what is happening to the efflux? Yes, when atorvastatin is there, then cholesterol efflux is obviously reduced using body efflux cholesterols. So if you increase the cells, stimulate the cells with increasing concentration of cholesterol efflux is increased, obviously. And so in spite of ABCA1, our question was, in spite of downregulation of ABCA1, how cholesterol efflux is increased? So it could be because of ABCA5 is there. So you can silence ABCA1 in the cells. When ABCA1 is silenced, ABCA5 is increased. Similarly, when ABCA5 is silenced, ABCA1 is increased. And very interestingly, when ABCA5 is silenced, you can see that most of the cells are going towards the late apoptosis, necrotic as well as apoptosis. So it is the signal for the necrotic plaque, advanced plaque. So when ABCA5 is also decreased, so the plaque become necrotic. That is because of most of the cells are become necrotic. So in the late apoptotic. So live cells are not very large in number. So now we try to validate the last part in the in vivo system, the knockout mice. In the knockout mice, we have used for this we have used two kinds of knockout mice, apo-knockout mice and pre-alpha knockout mice. We have fed the mice with high cholesterol, and we have checked the arterial lumen. You can see the lumen is almost closed, so there is a huge plaque deposited in the artery. You can see this normal chow diet, and this is because of the feeding the animal with 2 months with high cholesterol. You can see the high cholesterol diet, the plaque is almost the advanced plaque stage. And in this case, we have checked what is happening in the status of ABCA1. ABCA1 is decreased, ABCA5 is increased in PPR alpha knockout mice as well as in the apo knockout mice in response to cholesterol feeding. So high cholesterol diet, ABCA1 is decreased, ABCA5 is increased. High cholesterol diet after 60 days, ABCA5 is increased, but ABCA1 is decreased. So whatever data we have found in the human samples, the patient samples is reproduced in the animal cell culture and also in the animal model. And then we checked in the human patient, again, not patient, in the human asymptomatic individuals, the normal, so-called normal individuals. So in the normal individuals, we can divide the individuals into 4 groups. They are based on the plasma cholesterol, the low cholesterol and high cholesterol group. Among the low cholesterol group, we have divided into low HDL and high HDL, and also high cholesterol group, low HDL and high HDL. You know, there is a common, most of you know that high HDL is good for the system because high HDL will remove the cholesterol by reverse cholesterol transport. But even in spite of high HDL, high LDL, if they are high cholesterol, if HDL is high, it is good because it can manage the cholesterol level. But low HDL and high cholesterol level is dangerous. So that is this G2 group. So what is happening? These transporter proteins in this in human sample, the so-called. Yes.
Yeah, another 2 minutes.
Yeah, it's almost done. Yeah. One more slide.
Okay.
So you can see that in the high cholesterol group, ABCA1 is decreased, but ABCA5 is increased very significantly. It is also true in other groups. So whenever cholesterol level is high, the ABCA5 is there to rescue the system. So what is happening? Are they structurally different? So now we have done we have collaborated with the computational biologists who are doing the structural biologists also. They do a lot of structural simulation, and we ask the question: what is the similarity between ABCA1 and ABCA5? Because ABCA1 is known to be cholesterol transporter, but ABCA5 is not known to be cholesterol transporter. We found first time in our study. So he did a lot of simulation in cultivation. So, and after a lot of simulation study, what we have found, very interesting observation, that yes, this is the active transporter of cholesterol, the ABCA1. It behaves like ABCA1. Its structure looks like the transporter proteins. It's a transformer domain and other structure, and it helps the cholesterol from transfer the efflux from the cell inside the cell to outside the cell by active transport process with the help of HDL. But this is the secondary transporter, the ABCA5. The ABCA5 is not open. It is very closed state. It is not open. It opens only when there is a huge amount of cholesterol in the blood. So under physiological doses of cholesterol, ABCA1 is the primary efflux mediator by energy-dependent process, while ABCA5 acts as the secondary transporter. Under excess cholesterol, downregulation of lipid metabolism results in reduced role of energy-dependent ABCA1 for cholesterol efflux, thereby promoting ABCA5 as the primary mediator, and it is because of the gradient-based efflux. whereby it adapts to gradient-based efflux mechanism by conformational change. So its conformation is changed in such a way that it gives it this side is open and it gives passage to cholesterol from inside to the outside the cell. This is helps in the efflux procedure. So this is a very novel mechanism. First time we have discovered and is validated in the 3 different systems: human patient, cell line, and the animal model. So this is published in Journal of Molecular Biology this year, 2020. And this year, 2021, we have a cover page article in MCB, January issue. If you open the January issue, you'll find the paper is our paper. This is not regarding the cholesterol metabolism, but this is regarding the cardiac hypertrophy. And we have found, discovered one interesting regulatory mechanism of PP alpha signaling cascade by using the stimulation mechanism, which is published in MCB, and our figure, the mitochondria structure is our figure, is shown as the cover page article. So thank you very much. These are my groups. So these are the people who is involved in the human sample work. Olipto did most of the ABCA5 work, and Opamrito did the proteomics work. Deosmita is still working, and Prithushti is also working. They are working in the human sample still now. This group is working cardiac hypertrophy in the animal model. Ritu and Dibanti published the paper in MCB this year. So she's also leaving for America next month. And thank you very much. This is Professor Swami Bhattacharya. This picture was taken in 2019 in our IASF program in Kolkata. And this Kamalika postdoc, she was postdoc, she did a lot of proteomic study, and Trina has joined recently in my lab, probably the last student in my active PhD career, research career. Thank you very much. Thank you.
Thank you.
Share this slide.
Thank you, Professor Bandyopadhyay. It was a fabulous talk, and you have shown that how much work your lab has got into this. It is really a remarkable achievement to get a cover page in MCB, and definitely so on and so forth. And now the floor is open for a quick question. I can allow only one quick question because we are already, I want to stick to the time. So I request.
Thank you very much.
Okay, it seems nobody has any question as of now. I have one question, sir. What about the giving niacin? In US, they treat the high cholesterol patient with niacin. So, do you have any idea about that?
No. Yeah, we don't work on the treatment strategy. There's a clinical
They give the niacin is basically a lot of vitamin C. They pump into patients who are having high cholesterol with MI.
Yeah, that actually basically works in the oxidation mechanism, oxidized Ldl. As you know, that I have shown, and many other people have shown, that it is the oxidized Ldl which is the culprit. The oxidized Ldl is cleared by HDL into the macrophage, and then macrophage become foam cell. So if the oxidized Ldl is low, the cholesterol level will not be a problem. Even the cholesterol level is high. So in many patients, we have seen that oxidized LDL cholesterol level is high, normal, but there is a heart attack. So we checked those patients in the laboratory to check the oxidized LDL level. We have seen the patient with normal cholesterol but heart attack. Most of the patient, we have seen that oxidized LDL level is high. Normally in the clinic, they don't do oxidized LDL assay. So they give us the cholesterol profile, but they don't give oxidized LDL profile. So what we do, we bring the sample here and we check the oxidized LDL in our laboratory.
Okay.
So probably the vitamin C works nicely in many people have shown, because the oxidized LDL will remain under control.
Okay, thank you. Professor Roy, I guess he has raised the hand. Over to you, Professor Roy. Yes, Professor Roy.
He's mute.
Professor Roy, you are mute. Can you unmute? And
What about the EPA, max EPA? Because a lot of work has been studied with the max EPA and whether the max EPA can able to change this regulation ABCA1 or ABCA5.
And I don't have any idea, max ever. So first we show that okay, the ABCA5 is increased and it helps in the removing cholesterol, but not to certain extent, of course.
Okay. Thank you.
okay, so I take the opportunity to thank Professor Arun Bandyopadhyay for his excellent talk. Thank you.
Thank you
So-
very much. Thanks to all for listening. So I'll now leave. I have to go to other campus.
Okay.
Thank you very much.
Thank you. So now I request.
Thank you all for joining us.
So, due to the constraint in the time, I will request the other eminent speaker, Professor P. Prakash Babu, to upload his presentation, and Dr. Shamshad Sheikh to introduce him. Thank you.
Yes, sir. Thank you. Our second plenary speaker for today is Professor P. Prakash Babu. Professor P. Prakash Babu is the Dean of School of Medical Sciences, University of Hyderabad. He received Masters from Andhra University and PhD from University of Hyderabad. Subsequently completed postdoctoral training from Japan and University of London, UK. His main research interests are neurodegenerative diseases, cerebral malaria, glioma, meningioma. He has guided 20 doctoral students, 4 postdoctoral students, and is currently supervising 8 PhD students and 3 postdoctoral students. He has published more than 100 research articles in the peer-reviewed journals, presented his research in more than 140 national and international conferences, and has also organized many conferences of national and international significance. His publication citations are more than 2,600, and H index is 226 and I-10 index is 52. He has served as the Pro Vice Chancellor to the University of Hyderabad and currently serving as the Dean, School of Medical Sciences, University of Hyderabad. His prominent administrative positions in the University of Hyderabad include Dean Students Welfare, Chief Proctor, Head Department of Biotechnology and Bioinformatics. He has served and is serving across various important committees in the university, biotech and pharma industries, and hospitals in different capacities. He is Fellow, National Academy of Biological Sciences, Chennai, Fellow AP Academy of Sciences, Fellow Indian Academy of Neurosciences, Lucknow, Secretary Asia Pacific Society for Neurochemistry, Singapore, General Secretary Society for Neurochemistry, India, Secretary General Federation of Asian Biotech Association, Vice President India Society for Parasitology. Currently, his lab has grants from DBT and DST. He has active collaboration with KIMS Hospital, Dr. Reddy's Institute of Life Sciences. He's a chairman ICSCR Asian Institute of Gastroenterology, member ICSCR LV Prasad Eye Institute and JIPMER Pondicherry. He is member Research Advisory Committee, Nizam Institute of Medical Sciences, member Scientific Advisory Committee, ICMR, National Animal Resource Facility for Biomedical Research. Over to you, sir.
Very good morning, and thank you for your kind introduction. I thank the organizers Professor Rambala Roy, Shanti Desai, for inviting me to interact with all of you. And also I thank the other organizing committee members, and I also thank the chairpersons of this committee, this session. I congratulate the university and Department of Zoology for organizing such a wonderful meeting with all covering all the life process and all. So with this few words, I will take my talk, and I will be talking very briefly, of course, not going very deep into the subject, so that students may not really like it. And also my main aim is to let the students understand, and also to students to get some kind of inspiration out of the talk. So let me go ahead with my slides. Hope my slides are visible.
Yes, sir.
Right. So this is my talk of the today's my talk. So translational research. How exactly the research from lab to the bench to the bedside. So unless any life scientist does some work which is translated for the human well-being or the health sciences, so that may not be really good for the good idea. So to complete one particular aspect. So therefore, keeping that in mind, my lab has been working in this area. So one of the areas I'm going to present. So the basic, normally the basic research is a very essential thing, so that we can identify the fundamental processes, fundamental knowledge, fundamental mechanism of how this at cellular level it is happening. So unless we know this basic processes, you cannot go for the applied part of it. So this is the whole era of the translational research. So the basic research should go to the translational research. This translational research should go to the clinical side. So this is the basic essence of my talk. Now the major steps in the translational research is so basic research should go to the preclinical development. Then it goes to the clinical trials. Then it is going for the treatment purpose. Now, this how the applied aspect is directed towards the specific goals. It discovers the new medicines or new entities or new drugs or medical devices or the surgical procedures. So that will elevate elevate the human suffering. So there are so many diseases, as earlier speaker talked about the cardiac care. Now, the preclinical studies includes the in vitro research, as well as the in vivo research. So these two things are involved in this. So my lab is working with the cell cultures as well as the animal model, and also from the human samples that are obtained from the hospitals. These are the research areas that my lab has been working. I have the research animal model in the cerebral malaria, which I'm not going to talk now. Cerebral malaria is the malaria when it reaches the brain, it damages the blood-brain barrier that causes the cerebral malaria. It has a lot of symptoms that also causes the cognitive deficit in the children especially. There is one more called as the meningioma. It's a tumor or the cancer to the meninges. And the other one is the glioma, which I'm going to talk about today, epilepsy. And also we are also working with the cerebral ischemia, it's a stroke, which is a paralysis. So the other one is the stem cell therapy, how the neuroregeneration can be done using this. Now, as you know, the brain is a special organ in the body. It's a very special organ in the body. So there are a lot of things which, though the scientific advancement could reveal a lot of things in the brain, but still a lot of things are unexplored or unknown. So therefore, a lot of research is essential in this brain research. Now, this is the simple anatomy of the brain. So for this benefit of the students, one can see the frontal region, parietal region, occipital region, temporal region, and below is the cerebellum. So if you see the from the top side, the brain is divided into two portions, two exact halves. So and also the central sulcus that makes the brain into two frontal and the parietal regions. and also the occipital region. So these are the principal anatomy. And if you see the different regions again further, so prefrontal area is there, which is every region of the brain, every small inch of the brain is essential because it has some function. Even in the neurosurgeons, when they are removing the small part of the brain, they think several times how exactly whether this can be removed or not, because that function will be lost. Even in the brain tumors, when they are removing the tumor part, they will be very cautiously they remove without affecting the adjacent areas. So that is such a sensitive organ it is. Now I will try to project one video for the benefit of the students. If that works well, then it would be nice. Then let me see. Are you able to see?
Yes, sir.
Cerebrum with the brainstem attached and the cerebellum removed. The functions of the cerebrum include the senses of vision, hearing, smell, touch, and spatial perception, and also speech and language, memory, thought, and voluntary action. The cerebrum is formed mainly by the two cerebral hemispheres. These are separated in the midline by the falx, which occupies this longitudinal cerebral fissure. Though they look hemispherical from in front, the shape of each cerebral hemisphere is more complex when seen from the side. In front, this part, the frontal lobe, occupies the anterior cranial fossa. This part below, the temporal lobe, occupies the middle cranial fossa. This part behind, the occipital lobe, lies above the tentorium. The two cerebral hemispheres are connected across the midline by the corpus callosum, which runs all the way from here in front to here behind. The two cerebral hemispheres are connected below by the two cerebral peduncles converging on the brainstem. They're also connected by the structures of this area, the floor of the third ventricle. To see these connecting structures better, we'll look at a brain that's been divided in the midline. Here's the corpus callosum. This is the cerebral peduncle. The third ventricle is here. Here's the third ventricle in the model. It's quite narrow from side to side. This is the floor of the third ventricle. The surface of each cerebral hemisphere is richly folded. Each inward fold, or sulcus, it's very deep. It extends all the way around to here on the underside. Here's the medial end of the lateral sulcus in the intact brain. The lateral, it's the only one that runs all the way to the medial surface of the hemisphere. The cerebral hemisphere is described as consisting of 4 lobes: the frontal, temporal, and occipital lobes that we've mentioned already, and the parietal lobe. Between the frontal lobe and its neighbors, the central and lateral sulci form natural boundaries. The other boundaries are somewhat arbitrary. Here are the 4 lobes on the medial surface: frontal, parietal, occipital, and temporal. The sloping underside of the occipital lobe conforms to the upward slope of the tip of the temporal lobe is the uncus. The uncus lies just above the tentorial incisure, which is here. The olfactory nerve emerge. Body, a posterior horn, and an inferior horn. The anterior horn is in the frontal lobe. The body is in the parietal lobe. The posterior horn is in the occipital lobe. And the inferior horn curls downward and forward into the temporal lobe. To see where the lateral ventricle communicates with the third ventricle, to see how the lateral ventricle the third ventricle, and the fourth ventricle are connected, we look at the vascular tissue in the roof of the ventricle is the choroid plexus, which produces cerebrospinal fluid. Here's the interventricular foramen opening into the lateral ventricle. The choroid plexus passes through the foramen and continues into the lateral ventricle. The cerebrospinal fluid that's formed in the lateral and third ventricles passes through this narrow passage, the cerebral aqueduct, and into the fourth ventricle. Fluid leaves the fourth ventricle through three openings: the lateral apertures, the right one is in the depths of this recess, and the medial. Here it is, in the inferior medullary velum. The lateral openings are here. The medial opening comes out here, between the cerebellum and the medulla. The lateral opening on each side, large space surrounding the brain and spinal cord. We'll see where the cerebrospinal fluid is absorbed later in this tape when we look at the blood vessels. We'll return now to the underside of the cerebrum to look at the structures that form the floor of the third ventricle, which is here. Here's the optic chiasm. Behind it, this tubular structure that's been divided is the infundibulum, the stalk of the pituitary gland. These two projections are the mammillary bodies. To see how the pituitary gland is attached to the brain, we look at an intact specimen divided in the midline. Here's the floor of the third ventricle. Here's the optic chiasm. Here's the infundibulum, leading down to the pituitary gland or hypophysis. The anterior and posterior parts of the pituitary gland are quite distinct. The pituitary gland sits in the pituitary fossa. The pituitary fossa bulges downwards into the roof of the sphenoid sinus. This area just above the pituitary stalk is the hypothalamus.
Hope now students must have got some kind of idea about the brain and its morphological structure, morphology or anatomy. See, there are brain tumors. This is not one brain tumor. There are so many brain tumors are there. Many types of brain tumors are there. So which we are not going to touch upon. So today's my talk will be completed in another 15-20 minutes. Is the glioblastoma multiforming. It's also called as the glioma or GBM. So this is used to describe the different types of the glial tumors again. So astrocytoma, oligodendroglioma, ependyoma, and glioblastoma. So they are also can be divided into low grade and the high grade. So if you see in the picture, you can see in the right side of the human brain, it's called as in the frontal lobe. Just glioma basically it is seen in the cortex. It is in the cortex, especially in the frontal area. So this is the below one is the MRI picture of the glial tumor. Now the brain is having the different types of the cells. The major cells are the neurons and the astrocytes. It's called as the glia. So the neuron is the one which is actually responsible for all our cognitive or memory, whatever we are doing it. So, but whereas the other cells are called as the glial cells, these glial cells are nothing but the supporting cells of the neurons. The neurons they cannot be divided, they cannot be regenerated. If something happens to the neurons in the later stage, when somebody suffered from a stroke, paralysis, when that particular area is been damaged, so that particular area neurons are dead, glial cells also will die, but whereas the glial cells may come back, but not the neurons. Therefore, a person suffered with a paralysis, he will not recover, basically. So glial cells, they can be regenerated. So within the glial cells, there are called as the astrocytes, then oligodendrocytes. These oligodendrocytes helps in the myelination. If the oligodendrocytes are not appropriate, then the demyelination diseases will come. So these are the astrocytes are basically supporting cells. The glial cells all these glial cells are actually supporting cells to the neurons. So now the whole problem comes. Normally the glial cells, they keep dying, they keep regenerating. So if the 100 glial cells they die, 100 glial cells should be regenerated. By any chance, if the 100 glial cells they die and thousands of the cells are regenerating, then that is the problem that causes the glioma. So the cellular origin of the glioma, it's when there is a neuronal progenitor cell. From the progenitor cell, it comes as the mature astrocyte. These astrocytes will divide into give rise to the different cells. But whereas this transforms here during differentiation and give rise to the tumor astrocytes. Now there are two pathways we can see that one. So normally the glioma, this tumor. So normally any tumor, the liver cancer or the kidney cancer or intestine, whatever it is, all cancers, they grow from the 0 to the 4th stage. Grade 1, grade 2, grade 3, and grade 4. So here also in the case of the glioma also, it goes to the grade 1, grade 2, grade 3, and grade 4. This is the normal usual phenomenon. So glioblastoma is the deadliest of all the tumors. It's called as the deadliest of all the tumors because two reasons. One is there is no drug as of now which can cure the glioma. Second one is it will not give much time to the patient. Not more than 14 months, 13 to 14 months. So the patient survival is very, very difficult, very, very difficult. So that is the reason this is called as the deadliest form of the glioma. Why it is called as the deadliest form of the glioma means because here it is shown in the picture, the right side panel, it shows the glioma, it's astrocytoma, goes into the first stage, second stage, and third stage, and ultimately it goes to the 4th stage. It's called as the secondary glioma. The primary glioblastoma is this has a unique feature. From grade 1 straight away it jumps to the grade 4. Grade 1 to it jumps to the grade 4. That's why this has become a deadliest of all the tumors. So when it reaches the grade 4, as you know, it must have been metastatized. Here it is expressing the how the pathway is. So from the astrocyte precursor stem cells, it goes to the low grade astrocytoma. It's called as the World Health Organization. They have given grades. It's a grade 2. Then followed by the anaplastic astrocytoma. It's called as the grade 3. Then it goes to the secondary glioblastoma or the grade 4. Whereas in the case of the left side panel, it goes directly from grade 1 to the grade 4. So these are the genes that they are indicated, which are being elevated in the final 4th stage of the glioblastoma multiforming. Now, this is how we can categorize them glioma. So there are some oligodendroglioma. So when the oligodendrocytes they become a tumor, it's called as the oligodendroglioma. When astrocytes they become a tumor, it's called as the astrocytoma. When the ependymal cells, which are the actually as you have seen in the ventricles, ventricular lining is called as the ependyma. So these ependyma cells also will get proliferated disproportionately. That also gives rise to the tumor. Now, World Health Organization divided into grade 1, grade 2, grade 3, and grade 4. Grade 1 and grade 2 are called as the low-grade tumors. Grade 3 and grade 4 are called as the high-grade tumors. So low-grade tumors can be treated up to some extent, but not at the 3rd stage and the 4th stage. Now, some of the symptoms are, if they are having a tumor in the brain, so they will start vomiting. So mood swings, cognitive decline, hearing problem, headache, speech problems. People will suffer from seizures and all. Now, so these are the grades that one is the tumor is called the polycystic grade 1, grade 2, grade 3, and grade 4. Grade 1 is normally seen in the case of the children, and this is mostly curable. Then grade 3 is seen in the age around 35 years. So it gives lifespan of 10 to 15 years. Grade 3 is in the age group of 41 years. It gives lifespan of 2 to 3 years. Grade 4 is above 55 years. It gives not more than 1 year of the lifespan once it is diagnosed. Now, these are how the tumors are being graded into different types. Now, so the grade 4, the glioblastoma is poorly differentiated. High mitotic index. Mitotic cells are very, very high, and it's a diffuse invasion. It invades into the entire brain. Angiogenesis is seen in all these tumors. Then pseudopalletizing necrosis also can be seen in this. So these are the genes that they are like RB1, CDK, EGFR, PGFR, all these P10, KRAS. All these are the genes that they are getting overexpressed. Now, if you see this patient, so patient was MRI in April 2008 to 2008. So you can see the tumor, but whereas in the after post-operative May, so again it recurred. Again, it recorded. In the second case, in the August, they diagnosed with the tumor, and once it is again in the July 2009, again it has recurred. So it's a recurred into a large. So second time, normally they don't want the surgery, and person will die. Now, the glial tumors also, there are so many glial tumors are there. There are many varieties of the glial tumors are there. Now, what are the possible treatments? Surgery is the best option. Surgery should be done. Then radiotherapy will be given. So then chemotherapy will be given. So then also molecular targeted therapy or the monoclonal antibody therapy is also being given. So normally temozolomide is the one which is normally preferred treatment for this glial tumors. So the survivability after surgery, person may survive up to 4 months. Surgery plus radiotherapy, person will survive up to 9 months. Surgery plus radiotherapy and chemotherapy, person may live up to 12 to 14 months or 15 months maximum. So these are some of the genes that they are going to express in all these. So many genes are there. So many pathways are operational in the case of these tumors. Now, exact cause is not known. So people may attribute that there is a radiation exposure or some genetic factor or disorder of the immune system or somebody exposed to the different varieties of the chemicals or the viruses. So these are the possible causes. There's no pinpoint one can say that this particular cause is responsible. So the current standard care is the, though we have been giving all these treatments, but still we are yet to do it. So therefore, the current research is essential to identify where the mechanism can be blocked, or addressed so that the treatment is possible, and what are the drugs that can be given to the patient. So in this one, so normally the animal models, apart from the cell cultures, we should also work with the animal models. The animal models can be, so glioma can be induced chemically or xenograft, and also genetically injured. These are the 3 models are available. So we are working with the chemically induced one as well as the xenograft models that we have been working in our lab. So this is the chemically induced one. This is called the ENU, ethyl nitrosourea. If you inject into a pregnant rat, pups born are having the brain tumor. So the pups will show the corneal erosion, white loss and fur loss, and also depressive, or some are become very aggressive. So this is how we can make a model. So once after making the model, you can see the sections of this brain, whether they develop a tumor or not. Ka67 is the marker for the GBM, Glioblastoma Multiforming. Then this model is actually exactly mimicking with the human glioma. Therefore, the signaling pathways like c-MYC, n-MYC, and cyclin D1, these are all the positive for this tumor, indicating that this is similar to that of the human glioma. And we wanted to work with the different drugs, how these drugs can be used for this. So one is the repurposing of the existing drugs. So existing drugs, whether they can be used for the tumor, targeting the tumor. So when you see the tumor microenvironment that is inside the tumor is always the acidic in nature. Because of its acidic in nature, so drug resistance is developed, the existing drugs are not working. So if you neutralize that tumor microenvironment, so then there is a possibility of the treatment, better treatment. So what we have used is initially we used the rabiprazole, pantoprazole. All these are normally we have been using these drugs as an antacids, not antacids. It's they reduce the acidity, acidic in nature. So they are actually proton pump inhibitors. They accept the H+ ions, therefore it will become neutralized. So therefore, we used this rabiprazole, especially particularly we started with the cell cultures. When we used the cell cultures of the glioblastoma cells, so these cells, they showed that there's a promising results that it is inhibiting the cell proliferation. Then, so this is how we used it at different concentrations in the buffered and unbuffered medium. In the unbuffered medium, we have seen the best possible results using the rabiprazole against the C6 glioma cell lines. Then these cells morphology has been changed. That is indicating that these cells might undergo apoptosis. So this can be used to reduce the tumor cell load or prevent the cell proliferation or induce the cell death. Then when we checked with the FACS analysis also, it is pushing towards the apoptotic mode of cell death. And also when we studied the apoptotic proteins like PARP has been cleaved. PARP cleavage is an indicator of the apoptosis, and caspase 3 is elevated. Caspase cleavage has been done. So that is indicating the final stage of the apoptosis. So the cells are here. You can see also some of the cells using the DAPI and tunnel. So tunnel positive cells are seen in this using the rabiprazole. That means the rabiprazole also inducing the apoptosis. And this is compromised with the cell cloning, say cell cloning and cell migration. So these cells are, if you see in the bottom one, that is the unbuffered medium, you can see at the 200 micromoles, the coloning of the cells. Colonization of the cells has been reduced. Sorry, it's not cloning, it's a colonization. Colonization has been reduced. And bottom one is the wound healing. Wound healing refers to that of the metastasis. Metastasis are the migration of the cells. Migration of the cells has been reduced by using the rabiprazole. That means metastasis can be reduced by using the rabiprazole. And this is how the normally the EMT pathway is there. One the cells that from EMT to transition to the mesenchymal or mesenchymal to the tumor cells. So there also this is preventing its transition. We have done extensive work on this and published it, but I am not exactly completely using this one now. Now the anti-cancerous efficacy of the rabiprazole in C6 gliomas also. So in the rat, as you have seen in the brain we made a burr hole in the rat. So injecting the cells, the tumor cells, into the ventricular region. Once it is injected into the ventricular region, living for a few days, this develops a tumor. So then using if you see the graph, so normally vehicle treated one, animals will die. Animal will die at the around 26-27 days. But whereas the rabiprazole treated animal, it survived. So the even tumor size also reduced when it is treated with the rabiprazole. And histopathology also indicating that, so this is also, rabiprazole is also mitigating the tumor positive cells using the Ki67 and the STAT3, indicating that the rabiprazole can be used as one of the effective treatment strategies for this one. And so then we wanted to see that whether the plant-based drugs also can be used for treating this one. So this is the paclitaxel is the normally seen in all the gardens or anywhere. Actually, it even grows everywhere. Even this is also called as the graveyard plant. Normally in the graveyard, so many plants are there. From this, paclitaxel is an alkaloid. Alkaloid has been prepared from this plant. It's called as the paclitaxel. So this has been used against this one. So we induced the tumor again. So the tumor, we injected this paclitaxel packed nanoparticles. When the nanoparticles are injected into the rat, the tumor has been regressed. That is reduced in its size. So though we have done a lot of molecular mechanisms, I am not presenting completely. Now, other plant-based one is the Plumba Gene, is another natural plant. So from this, there is a plant is called as the Plumba Geneaceae family, and this is the compound is called as the Plumba Gene. So this is also isolated and it's available. So that also we worked with this, and this is also able to. This is a natural compound, so this is also able to reduce the tumor size. This also indicating that the this tumor morphology, cellular morphology has been changed, and this also changing the mitochondria. So mitochondrial membrane has been depolarized. Once the mitochondrial membrane is damaged, so that releases the apoptotic factors like cytochrome C that causes the apoptosis. So we published this also, and tunnel indicates. Yes.
Professor Prakash, you have another 2 minutes.
Exactly. I'm going and ending also.
Thank you.
So this tunnel positive cells also increased in using the Plumba gene, indicating that this is pushing the cells into the cell death, apoptotic mode of cell death. So this Plumba gene also, when we studied in the rat model, so we have shown that the Ki67 and VEGF has been reduced when Plumba gene has been used. So that indicates that again, so this is also able to control the tumors. So other one is the xanthosylene nitidine. So this is also a plant-based medicine. So this also indicating that, so this is PDOC, when we have done with the PDOC antibody, so that is reduced at the different time points. So 8 days, 4 days, 8 days, and 12 days. So that is indicating that this also can be used as an effective drug for the treatment of this one. So the altered cell cycle also has been seen in the, so that means the cell cycle is going to inhibit and also inducing the apoptosis when we studied with the FACS analysis. So the apoptotic, most of the apoptotic proteins are elevated, indicating that this is pushing towards the apoptosis. Now, in our conclusion, so these, though we are able to work and show some of the drugs that they are able to control, but further studies are essential to take to the next level. This is a preclinical studies, and it has to go for the different stage, a few more studies to go into the final stage. So incidentally, tomorrow is the Glioblastoma Awareness Day. So therefore, I have chosen this topic to present what are the little that our lab is doing and published for the last several years. So this is my lab group. So some of them, they are in US right now, those who have completed, and below 2 people are neurosurgeons who are associated with us. So thank you.
Okay, so another excellent presentation. I'm sure many students have liked it, and with the kind of topic Professor Prakash is dealing with, it is very interesting. So if anybody has I can allow 2 questions on this. Probably one person has already written a question.
So the question is in the chat box.
Yeah, I'm going through that. Yeah, in this model, apart from the acidic nature of the tumor microenvironment, so there is a possibility of the hypoxic condition. So these two are actually the culprits for the drug resistance. So we have to address both of them at the same time. So vasodilation is not the one actually. So when there is a tumor microenvironment change, there is a necrotic mode of cell death will be there. So that actually causes the edema and the inflammation in the brain. So that is responsible for the ultimate final death of a patient because of the respiratory failure. So that has to be addressed. Yeah, so actually that video is good, but whether you observed it or not, I forwarded that video due to lack of time. So maybe next time when there's a possibility, definitely I will show the complete brain, different regions and their functional anatomy, and I will choose different topics. So maybe Dr. Shanti Desai can take initiative.
Excuse me, sir.
Yeah, please.
Yeah. So with regards to that question, it was asked without a tumor condition or in the absence of a tumor under normal physiological conditions, if you are trying to induce hypoxia. So in that case, the somewhat the blood flow reaching to the target organ, or here in this case the brain. So when we try to induce hypoxia, astrocytes will counter that by vasodilation, but which will increase, which will lead to a normal blood supply for a limited period of time. So how do we counter that?
No, astrocytes hypoxia, where is the induced vasodilation? Why do you think that astrocytes will? So it will not increase the vasodilation. It creates the angiogenesis. Actually, whenever there is a hypoxic condition or there in any organ, it causes the angiogenesis, neovascularization.
Thank you.
So wherein what one has to use is the VEGF treatment, vascular endothelial growth factor treatment, that should be withdrawn.
Okay, so excuse me. Like if anybody has any specific question, they can send to Professor Prakash. I'm sure he will be able to reply. We'll go. Thank you very much, Professor Prakash, for your time. As we are running short of time, I would like to go for the next presentation. I'd like the reporter to introduce the next speaker. Thank you.
Yeah, thank you, sir. The next speaker for today is Dr. Ashwini Kumar. Dr. Ashwini Kumar is the director of ICMR Vector Control Research Center, Indira Nagar, Puducherry, India. He is university gold medalist and PhD in entomology. He has 34 years of rich research experience in vector-borne diseases. He has been trained in London School of Hygiene and Tropical Medicine, UK, German Center of Biotechnology, Braunschweig, Germany, WHO workshop in Myanmar on malaria burden estimation, as global organized course on science of malaria eradication, and advanced course on managing the end of malaria in Barcelona and Sitges, Spain respectively, and on vector control operations in 3 countries of California, USA. He successfully managed over 50 basic applied and operational research projects funded by ICMR, DBT, EST, NIH, WHO, World Bank. Among the major operational projects undertaken were the demonstration of malaria epidemic control in Goa, HIA of Konkan Railway Project, and vector control in Goa port using bioengineering methods. Dr. Kumar has to his credit 121 research articles and reviews in journals of national and international repute, including The Lancet and Genome Research. He has served as reviewer of manuscripts for several national and international journals and as editorial board member. He has edited 4 books, including a handbook for WHO, vector surveillance and control at points of entry, which is available in 8 languages. He is recognized by Goa and Manipal universities as PhD guide. Dr. Kumar has guided 15 PhD students. He has organized 9 national international symposia, conferences, and training workshops for capacity building. His international assignments include advisor WHO, expert on vector control tools for malaria elimination as global director, Society for Vector Ecology, and director, World Mosquito Association. Dr. Ashwini Kumar is recipient of 14 awards at national and international level, including Outstanding Young Person of the World in 1998 in Manila, INSA Royal Society Fellowship, NAVBT Biotech International Award in 2013, and Fellowship Award by Punjab Sciences Academy in 2018, among others. He has widely traveled internationally. He is a team player, a natural motivator, and believes in quality research through networking and team building. Over to you, sir.
Introduction. Thank you, Ms. Sheikh, for this wonderful introduction. And I must thank Department of Zoology, especially Professor Roy and Dr. Shanti for this kind invitation to make a small presentation before you. And I have a special connection with Goa University, as was alluded by Ms. Sheikh, because I was in Goa until 2019. And I know all the colleagues in the Department of Zoology. I had a very close interaction with other departments as well when I was researching in Goa. So with this, let's get to our topic. The topic of today that I'm dealing with is the One Health approach, a new approach for human well-being and sustainable development from a concept to reality. I would like to share my presentation with you, and for the sake of bandwidth, I will also switch off my video for the time being. Can you please see my screen?
Yes, sir.
Okay, so what is One Health? One Health is a collaborative, multi-sectoral and transdisciplinary approach working at local, regional, national, and global levels with the goal to achieve optimal health outcomes, recognizing the interconnection between people, animals, plants, and their shared environment. What are the UN Sustainable Development Goals? There are 17 of them, and the goal 3 says ensuring healthy living lives and promoting well-being at all ages is essential to sustainable development. WHO says currently the world is facing a global health crisis unlike any other. COVID-19 is spreading human suffering, destabilizing the global economy, and upending the lives of billions of people around the globe. COVID-19, as we know, we are passing through such a disastrous phase at the moment, is an outcome of coronavirus, which is a zoonotic in nature, and now it has caused havoc in entire world, as we know. These are the leading causes of human deaths worldwide. It's slightly older data, but it shows that about 15 million infectious diseases, infectious diseases cause about 15 million deaths. If you take the total number of deaths due to various diseases, and there are 12 different causes of infectious diseases listed on the right, which indicate that the respiratory infections are on the top, and followed by HIV, diarrheal diseases, tuberculosis, and now we are confronted with COVID-19 infection. Over a period of time, the NCDs, the non-communicable diseases have surged ahead of infectious diseases, communicable diseases, and we know that the cardiovascular diseases, diabetes, cancers now are surging ahead, far ahead of the infectious diseases, barring the infections that like now we are facing due to COVID-19, which is a pandemic. And let us see the history of these outbreaks. They say that if we do not remember the history, we are condemned to repeat it. About 100 years back, there was a major Spanish flu, and then various infections visited us throughout the last century. And if you would see, up to 2000, there were 10 major outbreaks, pandemics, and these were 10 in the last century, occurring at an interval of about 10 years, starting from Spanish flu up to the Nipah virus here. And if you see in the current century, there are already in 20 years 5 pandemics, which means their frequency is actually increasing at an alarming phase. So this slide summarizes the zoonotic pathogens and emerging diseases. There are close to 1,400 pathogens that are infectious to human, and 62% of them are zoonotic in nature. That suggests that the threat looms large from the zoonotic diseases, and out of 175 emerging diseases, 132, which means about 75%, are zoonotic in nature, and the world is so well connected. And of course, there is an interface between humans and animals, and this is increasing over a period of time. This slide summarizes the 3 kinds of emerging threats. One, the red dots, which are newly emerging infections, and the blue, which are re-emerging and resurging infections. And there is one black dot in the US, in the New York, which was, of course, was deliberately introduced, anthrax, to the New York, which was an act of bioterrorism. What does this show? That here there is one cluster in China where the new infections are emerging in this region over and over again. And in the US as well, if you see large number of the new agents, and of course re-emerging infections are becoming common over a period of time. If we break up the zoonotic infections, the largest threat to humans is from the domestic animals, and a much smaller threat from the peri-domestic wildlife, the captive, managed farmed wildlife, and the free-ranging wildlife. 99% of these are actually zoonoses in the red box, and these are acquired directly or indirectly from the animal reservoirs, which are in close proximity or contact with the humans. And the examples are rabies, anthrax, BSE, and Hendra virus. And then once the humans acquire infection, some of them may then set in from human to human, and these infectious diseases then cause the outbreaks and pandemics. And also, there is reverse zoonosis possible from humans to animals. For example, the Mycobacterium tuberculosis from humans to, and also there are some cases of cats or the wild cats suffering from COVID-19 have been also reported. So here is what it can be summarized, that there are close interaction between humans, livestock, and peri-domestic wildlife. And when these three overlap, there is a possibility of the pathogens being passed on from animals to humans. And there is a interaction between the wildlife and the livestock, and also the humans with the wildlife as the humans venture into the wilderness or nature, and overall the biosphere controls these interactions. This slide beautifully summarizes that what happens over a period of time when the humans, there are conflicts and natural disaster. Many of the disasters, as we know, are man-made now. Global warming is one, and the health systems insufficiency, the population growth, which is always on the rise, and of course mobility and transport due to globalization is increasing, thereby increasing the contact between the humans and the animals. And as a result of this, the environmental changes are pronounced. As the demand increases, the mass production, industrial production increases, it causes pollution, climate change, and of course urbanization and deforestation and travel and tourism. This is all about induced changes in the environment due to human action. And of course, as the demand for food increases, the food production, as far as animals are concerned, use of technology, farming practice, and the food demand increases. And of course, the food exports also increases. And this is a vicious cycle which goes on, and thereby increasing the pressure on the environment, and also increasing the chances of human and animal interface, and leading to the zoonotic infections. So again, what is the One Health definition? Let's visit that once again. This is collaborative, multi-sectoral, transdisciplinary approach. I think we are convinced now that we cannot, work in isolation or in silos. All the three disciplines have to come together, wherein the local, regional, national, and global levels, the optimal health outcomes can only be due to healthy interface between the people, animals, and plants, and their shared environment. So what is the impact of human action in the emergence of infectious diseases, zoonosis, antimicrobial resistance, and expansion, making One Health approach relevant? microbes keep on adapting themselves to the changing environment, and of course mutations do take place, and then mutations also lead to adaptability. Then the human susceptibility to infection over a period of time also increases. The climate change and the weather pattern changes also bring in these zoonotic pathogens in close contact with the humans through the animal-human interface. And the changing ecosystem, the encroaching of the forest ecosystem by the humans also leads to the same effect. And human demographic, the migration of human populations and their behavior also leads to these interactions. And of course, the economic development and land use pattern, which keeps evolving and changing due to economic development, also has the adverse effect on the ecological changes overall. The overall international travel and commerce, increase in technology and industrial output, as I mentioned, and the breakdown of public health systems, as we observed in case of COVID-19, a second wave in India, that how desperate people were even for the hospitalization and the oxygen requirement and all that. So health systems need to be strengthened. They are not geared up to meet any exigencies of the nature that we are facing with. And the poverty and social inequality is another reason, and war and famine, they also lead to the such kind of disasters and the breaking up of the health systems, and also the normal life that gets disrupted, and of course the lack of political will and the bioterrorism, which we all know is becoming a very big threat over a period of time. So how does this impact to the world? Zoonosis affects 2 billion people in the world and results in 2 million deaths annually, which is a very large number, as we can see. SARS, H1N1, Ebola infections cost billions to the world economy. Low and middle income countries suffered a lot, about $110 billion due to foodborne infections every year. Drug-resistant infections might contribute to a loss of $9 billion by the year 2050. And COVID-19 pandemic has already contracted, as we know, global economy by about 4%, and which means close to $4 trillion losses have already taken place. So this summarizes what is One Health approach. Here, I mean, this is borrowed from One Health Sweden. It beautifully describes individual health and the ecosystem health. They are intimately interlinked, and public health is linked with the veterinary medicine, the ecology, the environmental health. All these are under one umbrella: human medicine, molecular and microbiology, the health economics, all are interlinked. And if this is disrupted, this ecosystem is disrupted, it leads to various undesired outcomes as far as the diseases are concerned, both the NCDs as well as the zoonotic infections. So what One Health will offer? One Health approach will strengthen public health systems at the human-environmental interface. It will help to address disease risk and prevent the disease emergence at the source. One Health strategies are cost-effective, and it reduces morbidity, mortality, and economic costs of the outbreak. Here is an example in UP from the year early 2000 to 2014. It was suspected that in Gorakhpur area there was Japanese encephalitis as the major cause of the deaths in the children. But later on it was observed that the bulk of the problem was actually due to scrub typhus. 60% of that, the AES used to be acute encephalitis syndrome was due to scrub typhus, not due to JE. And although JE was also prevalent, and it was then found out that the animal hosts which were in the domestic and in the wild, they were crossing over and causing this infection of the Orencia gamushi, which is a rickettsial pathogen, as we know. And large number of cases were detected in that area, and this is an example of zoonotic interface between the wild and the local animals and the humans. Similarly, the mystery of KFT was solved by the One Health approach. We know that even in Goa, there is a KFD outbreak happening every year in the early months of every year now in Valpur area, and also in Dodamarg in Maharashtra. It used to be confined in Shimoga of Karnataka earlier, and later on this infection has spread both downwards towards Kerala and northwards towards Maharashtra and Goa. And this was solved because again the humans have been encroaching the wild environment, and in Valpur area there has been a lot of deaths of monkeys. Monkeys have contracted AFD, and when they die, the ticks leave these monkeys, and they can then cling on to the humans and can cause human infections. And there have been deaths. Normally during June to December, there is a zoonotic phase here, wherein the large animals, the ticks evict them, and then they reproduce, and then they normally unfed larvae will feed on small animals and monkeys. And later on, once these fed larvae are shed, and then in the month of January, February, then they can attack in the jungles to the humans to go for logging or other purposes, and also to the wild animals. This is how it is maintained in the nature. So what is the One Health initiative in India? What's being done here? India has enacted Food Safety and Standard Act in 2006. The Center of Zoonosis, National Center for Disease Control, has published a manual on handling of zoonotic diseases in 2006. National Standing Committee on Zoonosis was formed in 2007, and database of zoonotic disease research was prepared in 2017, and ICMR is currently establishing a center of One Health in Nagpur in Maharashtra. This is the last slide. It says that the future of microbes and mankind will probably unfold as episodes of a suspense thriller that could be entitled Our Wits versus Their Genes. Thank you very much. I stop here. Over.
Indeed, that was an exciting presentation. Now I can request others to, if they have any question. Yeah, Rahul, go ahead.
Hello, sir. Sir, I have a question. In 2016, Sri Lanka was declared a malaria-free country. So, is it possible for India to become malaria-free?
Thank you, Rahul. This is a nice question. India has also launched malaria elimination initiative, and the year for elimination has been kept as 2030. By 2027, India wishes to be malaria free, and it requires 3 years of no new infections for getting the WHO certification. And you may be happy to note that the incidence of malaria is reducing at a good pace in our country over the last 3 or 4 years, particularly in Orissa, which was hyper-endemic to malaria. The malaria in Orissa has more or less crashed, and that's the good news for India.
Thank you, sir.
Okay. So anybody else? If not, I thank Dr. Ashwini Kumar for the excellent presentation. And it was indeed, we got to know many things, that how India is progressing and overall scenario and the vector-borne disease management in India. Thank you very much, sir.
Thank you, sir.
So I'll request to introduce the next speaker, if she's ready. We are getting some time here.
Thank you, sir. I will continue. Next speaker for today is Dr. Manjula Das. Dr. Manjula Das is a senior researcher, MSCTR Bangalore. Dr. Das has been exposed to translational clinical research when she worked on making the first Indian vaccine against rotavirus during her PhD days at Indian Institute of Science, Bangalore. During her postdoctoral training, she was exposed to in vivo models for cancer and bioinformatics at McGill University, Montreal, Canada, CNRS, Rennes, France, and at MIT, Boston, USA. At McGill, Canada, she was trained under a clinical doctor, at France, a chemist, and at MIT, a mathematician, making her a truly interdisciplinary scientist. However, her training as a translational scientist wouldn't have been completed without her industrial exposure since 2001, when she has been working in the area of drug discovery and development in many companies in India. Her expertise has been in development of recombinant antibody and assay in vivo and cell-based platforms. She has been a serial entrepreneur for 20 years, being involved with 4 startups. In 2016, she came back to academia to utilize her expertise in product development in translating the research from bench to bedside in the area of molecular immunology. Currently, she leads a group of practicing clinicians cum researchers in the area of molecular immunology in Mazumdar Shaw Medical Foundation in the multi-speciality hospital in Narayana Health City, Bangalore, India. In 5 years itself, her interdisciplinary and collaborative efforts, along with scientific publications, have won her a number of government grants and patents. Since the wake of COVID-19, she has plunged into COVID research through publications, winning government grants for diagnostics and therapeutics, and of course, mass-scale patient testing through her clinical collaborators. She has been involved in developing RNA extraction-free direct diagnostic kit, an in vitro model for testing anti-SARS-CoV-2 molecules, and a novel fusion protein for the treatment of COVID-19. She collaborates with a pathology lab which tests more than 3,000 patients per day and a BSL-3 facility to do viral culture. So this was in brief about Dr. Manjula Das. Ma'am, over to you.
Thank you, Dr. Shamshad, for the nice introduction. Good morning, ladies and gentlemen and fellow scientists. I begin today's talk by thanking Professor Ramaballav Roy for giving me this opportunity to present my work my humble work in front of this distinguished audience, especially my previous two speakers. I thank them for their excellent presentation. I thank Dr. Ashwini Kumar for laying the background of this talk too, because you did speak about coronavirus, and the work that I'm doing, I'll take it forward through my presentation now. For past 1.5 years, many of us have sold our soul to virology. I was happy to come back to my first love, again, RNA virus, coronavirus, after doing my PhD on rotavirus, a double-stranded RNA virus. Anyway, I started working on SARS-CoV-2 last April, applied for a grant in May, and received the funds to develop a novel therapeutic in November 20. Here is to present what my team has done since then. Severe acute respiratory syndrome coronavirus, or SARS-CoV-2, is the third novel coronavirus known to us this century. And as we all know, it is the cause of the current pandemic. SARS-CoV-2 belongs to the class of beta coronavirus and has a positive sense of double-stranded RNA as its genetic material and is enveloped. The virus is characterized by its 4 structural proteins, and these are nucleocapsid proteins, envelope protein, the membrane protein, and the spike protein. The viral genome consists of 26 to 32 kilobase pair with 6 to 11 ORF. ORF1 makes up 67% of the genome and encodes for about 16 non-structural proteins, including papain-like proteases, chymotrypsin-like proteases, and RNase-dependent RNA polymerase, which is coded by ORF1b. The N and E are the canonical structural proteins coded by ORFs 10 and 11. The remaining ORFs code for other non-structural proteins and the structural proteins. Amongst the 4 structural proteins, spike protein is crucial for the virus to initiate the infection in the host. Spike protein can be divided into S1 and S2 domain, where the S1 consists of the receptor binding domain, or RBD, which is necessary for it to bind to the host cell receptor. The S2 domain comprises of fusion proteins, which facilitates the host viral membrane fusion. When the virus attaches to ACE2 molecule, which is the receptor, the protease present on the host cell surface cleaves the spike protein at its furin cleavage site and facilitates host virus membrane fusion, resulting in the entry of the virus into the host system. When there is viral infection, it naturally elicits an immune response in the host. First, the innate immune response is activated. This leads to the pattern recognition receptors, known as PRRs, to bind to the pathogen-associated molecular patterns, PAMPs, unique to the virus, which activates the immune response pathways, leading to the production of pro-inflammatory cytokines. However, an exaggerated response of the host immune system can cause various other complications, such as hypercoagulopathy and microangiopathy, leading to tissue damage, organ failure, and untimely death. So, during the viral infection, various cells, including the macrophages, the dendritic cells, T and B lymphocytes, express several transcription factors, including NF-kappa B, which further produces pro-inflammatory cytokines such as IFN gamma, IL-6, IL-1 beta, TNF alpha, which are the hallmarks of the SARS-CoV-2 infection. However, an overexpression of these cytokines result in clinically significant symptoms from high fever to tissue damage to organ failures. However, it is not easy to predict the severity in patients infected with SARS-CoV-2. The severity of SARS-CoV-2 is marked by the elevated levels of IL-2, IL-6, IL-10, TNF-alpha, and interferon. A meta-analysis study by our group showed that determination of levels of only these two cytokines, the IL-6 and IL-10, are sufficient to diagnose the severity of the infection. Let me see if I can get the... Okay, so by developing assays to detect these two cytokines, here we can see that it's only if it is only IL-6 or the IL-10, the combination of these two will be enough to predict the severity of the infection. So this was published recently in Helion by our group. There are various ways in which the viral infection is being mitigated now. So before the virus enters the cell, it can be the receptor and virus interaction can be mitigated by monoclonal antibodies. As soon as the virus enters, then the entry of the virus also can be mitigated again by different kinds of antibodies or inhibitors. The capping and other the immune mimicking behavior of the virus can also be mitigated by remdesivir kind of drugs. Then there are protease inhibitors which can stop the viral entry and viral replication. Similarly, there are other drugs which can be at different stages, which can stop the viral replication and the viral exocytosis also. So what we went ahead and did is that we made a soluble protein, a soluble ACE protein, which is the ACE2 is the receptor. I will talk a little more about it. We made the soluble ACE receptor, which will, before the virus enters the cell, it will go and bind to the virus, neutralize the virus, and wouldn't allow the virus to enter the cells. So little more about the angiotensin converting enzyme. Let me introduce you to the ACE molecule, to the receptor. The ACE2, the receptor molecule, it maps to chromosome X at a P22 position. It spans around 39.98 kilo base pair of genomic DNA and contains 20 introns and 18 exons. The ACE2 gene encodes a type 1 membrane-bound glycoprotein composed of 805 amino acid, which is a large molecule. The functional domains include the C-terminal transmembrane anchoring region, the N-terminal signal peptide region, which takes it to the membrane, and a zinc-binding metalloprotease motif. The ACE2 is highly expressed in capillary-rich organs like even nasopharyngeal tract, the different organs like kidney, and also in gut and brain. The physiological function of ACE2 is very important. The Renin-Angiotensin aldosterone system is one of the most important hormonal mechanisms in controlling hemodynamic stability by regulating blood pressure, fluid volume, and sodium-potassium balance. Renin is synthesized in the kidneys as an inactive form. and it released into circulation in response to low levels of intratubular sodium, hypertension in the afferent arterioles of the renal glomerulus, and sympathetic activation. In the bloodstream, prorenin is activated by proteolytic and non-proteolytic mechanism to produce the active form. Active form. Here, the active renin catalyzes the cleavage of the glycoprotein angiotensinogen, generating angiotensin 1. This Ang1 is cleaved by the angiotensin converting enzyme, which is ACE, which is different from ACE2, to produce angiotensin 2. The ACE2 is a critical enzyme in controlling any excessive vasoactive and growth promoting effects of the renin-angiotensin system. ACE2 catalyzes the conversion of Ang1 to angiotensin 1 to 9, which in turn can be converted to Ang1 to 7 by ACE. ACE2 also directly hydrolyzes the Ang2 to Ang1,7 with high catalytic affinity. Ang1,7 is a biologically active metabolite that counterbalances the cardiovascular effect of Ang2. Through the MAS receptor here, Ang1,7 exerts vasodilatory and anti-proliferative effects, therefore counterbalancing Ang2. So ACE2, this is just to summarize that ACE2 has a very active role in the normal activity, normal proper balance activity of human life. So if the ACE2 is impaired, which I mentioned that earlier days when they were trying to stop the viral entry, they developed multiple inhibitors of ACE2. So if ACE2 is inhibited, then excess of angiotensin 2 levels will be present, angiotensin 2 levels will be elevated, and thereby it will impair the balance of the RAS pathway, whose goal is to maintain a normal arterial blood pressure range by controlling water and electrolyte homeostasis. So angiotensin 2, the excess amount of angiotensin 2 will stimulate the production of aldosterone through the AT1R, which I showed in the previous slide, in the adrenal cortex. Through specific actions on the distal nephron of the kidney, aldosterone will promote sodium reabsorption, water retention, and potassium and magnesium loss, thereby modulating extracellular space volume and blood pressure, increasing the blood pressure. So since blood pressure is finely tuned by the RAS, any imbalance in this system will produce arterial blood pressure alteration. Aldosterone and Ang2 are the principal RAS molecules involved in the cardiovascular adrenal system, which changes during hypertension. Both molecules are also involved in the physiological control of blood pressure, directly impacting cardiomyocytes, kidney epithelial cells, and vascular smooth muscle cells. During hypertension, excess of these molecules have also been linked with cardiovascular and kidney tissue hypertrophy and fibrosis. So we cannot inhibit ACE2. We used the soluble ectopic region of the receptor. So what we did is we took the soluble ectopic region of the ACE2, and for increasing its pharmacokinetic activity, that so that the protein stays for a long time in the blood, we fused it to the Fc region of IgG, of the human immunoglobulin molecule. So, and the ACE2, which first we amplified the ACE2 from the human cDNA library, but what we saw that the ACE2 sequence is that it digests EcoR1, BAMH1, EcoR1, HIN3 And all these common enzymes also digest in the normal sequence which is available in human. So we introduce multiple mutations which will increase the binding of the ACE2 to the viral protein, as well as it will increase the production of the protein. So this mutated protein, the mutated recombinant protein, which we call solase2 for the soluble, human soluble ACE2, that was synthesized and it was cloned in PTT5 vector, and then it was expressed in HEK cells, and so it was expressed in HEK cells and the HEK293E cells, and we took the supernatant, and it was expressed in the supernatant, since we had put the HA tag, HA signal peptide, so that which takes it out to the supernatant in the mammalian HEK supernatant, and that by ELISA, we showed that ELISA performed with 1 microliter of the transfected soup. When we performed the ELISA with the spike protein, which we also expressed when we did the ELISA, we saw that like the expression was on till 120 hours. So and on SDS we could purify it, because we had also because of the FC region, it was purified on protein A columns, and the by Western also the by Western Western blot of the reduced solase2, and we could see a single band. We did the CD analysis of the purified protein, and the CD, as expected from the fusion of these two portions, the ACE2 portion and the IgG portion, that we see a mixture of alpha sheet and alpha helix and the beta sheet. The binding of the solase2 to the full-length spike was also shown by ELISA as well as by Western. When we wanted to check the activity of the protein in vitro platform, when we chose a platform for studying a molecule, it is important to pick an ideal one. In this case, we wanted to check it on Viro cells, because Viro cells were known to express the, as I earlier mentioned, that that kidney cells express ACE2. Viro cells are monkey kidney cells, and they express ACE2. So we wanted to use the Viro E6 cells for seeing the effect of the viral infection and our molecule, that how it prevents the viral infection. But Viro E6 cells, which express more ACE2, were better. And at that time, we also saw that another A549, which is a lung cell line, so that also came out, that which expresses more of ACE2. But when we wanted to use that, we saw that A549 ACE2 was unable to show the cytopathic effect, whereas Viro E6 could show the cytopathic effect. So that's why Viro E6 cells were selected for further experiments. So the experiment that on the in vitro platform, the experiment that we did was we standardized the antiviral screening by PRINT assay, which is a plaque reduction neutralization test with the SARS-CoV-2 infected with Viro cells. So when you grow only the cells, the Viro E6 cells grow like a mat. And when we put the virus enters the cells and it forms the plaque, which you can see here. So, and we standardized this assay with the help of a positive drug called remdesivir, so which I've also mentioned. So we did that in Viro E6 cells and with HUH7 cells. Both of them could show the cytopathic effect, though Viro E6 cells showed better cytopathic effect. We could see dose response with increasing amount of remdesivir. So with this, we went ahead and used our protein, the Solase 2 protein, for the print assay, and we saw that as a biomolecule, though we don't expect that the IC50 will be lower than this double-digit number, so we got very good IC50 with a biomolecule, which is a much larger molecule than the small remdesivir. And if you just see that the cytotoxic effect as expected is much lower as compared to remdesivir. Like in remdesivir, actually here, we have shown that cytotoxic effect was much more than what we expect from a biomolecule. So I'll show in later slides, I will show that we have done the in vivo assays also, where we see that the cytotoxicity is much lesser. So with this, now the entry of the virus to the cells were also seen by qPCR. We took the cells after infection, and both the Viro E6 cells, and there are some oral carcinoma cells also which we have used. I will talk about it a little later, that the Viro E6 cells and the M08 cells, when they were after infection, when we took the cells, sliced it, and did the qPCR, we see that virus had entered, and as compared to the uninfected cells. So here, at this point of time, I would want to switch gear a bit, and I would want to talk about the comorbidity of the COVID infection. As we all know that diabetes, obesity, and hypertension are well-known comorbidity factors of COVID-19 infection that leads to disease severity. However, there is a significant increase in severity mostly in hyperglycemic condition. Again, a meta-analysis study conducted by our group showed that increased inflammation with elevated CRP levels and loss of lymphocyte, as well as hypercoagulopathy associated with diabetes in COVID-19 patients, to be the determinant of comorbidity. As previously explained, the viral infection results in increased production of pro-inflammatory cytokines and modifications in NK cells, which further leads to vascular permeability for these cytokines, causing acute respiratory distress syndrome. Along with these, the increased ROS and upregulation of RAS result in insulin resistance, worsening a hyperglycemic condition, leading to vascular damage. This increases the clotting factors such as D-dimer in the circulatory system, making the blood viscous. All these events together lead to multiple organ failures and untimely death. Thus, it is important to have an in vitro platform to screen for therapeutic interventions, which can even decrease the comorbidity conditions like hyperglycemia. So in our team, earlier has developed a cell-based diabetic model in HepG2, the liver cancer cells. When the liver cancer cells were grown in normal glucose, in general, the HepG2 cells, as suggested by ATCC, it should be grown in 25 millimolar glucose. Instead of that, we grew the cells in 5.5 millimolar glucose condition and made the cells insulin resistance by giving hyperglycemic shock by growing them at 50 millimolar glucose. So glucose uptake was severely hampered in these cells, as well as increased ROS level and high accumulation of lipids. So the mRNA sequencing, we sequenced the cells grown in 5.5 and 50 millimolar. The mRNA sequencing results from these cells showed upregulation of various pathways, among which IL-17 and TNF-alpha pathways are prominent. To our interest, the level of ACE2 were also upregulated in hyperglycemic condition with no difference in, but there was no difference in the TMPRSS level, which is the protease which helps the virus to enter the cells, as I have mentioned previously. So for this study, what we did that for the comorbidity platforms, we took oral mucosal epithelial cells, which we developed in the lab. I will talk about it. And we took the lung epithelial, we took 2 oral mucosal epithelial cells. We took the lung epithelial cells, and we also took HepG2. So we took the oral mucosal cells to see the site of entry, the A549, the lung cells for site of infection, and HepG2 is like liver is one of the major organs where diabetes, the glucose homeostasis impairment happens. So that's why HepG2 was also chosen. So now a little bit about the oral mucosal cells, because typically oral mucosal cells are normal oral mucosal cells are not available with ATCC or in the market. So we also work with oral cancer patients. So from them we have for this study we have taken that from 2 patients, both 65-year-old female with no risk habits. They were diagnosed with squamous cell carcinoma, and M08 is one more patients, similar HNSCC patients, but it was diagnosed with moderately differentiated squamous cell carcinoma. These 2 cells, these 2 were spontaneously immortalized, and it could grow up to Passage 52. The growth medium was normal, our PMI 1640, but sorry.
Dr. Das, you have 2 minutes.
Sure, I'll just finish fast.
Yeah.
So when we grew them in 50 millimolar glucose, we saw that ACE2 expression in M08, actually M08 epithelial, even in M08 fibroblast, it increased. But if we took the M08 epithelial cells, and we saw that in M08 and B12, there was slight increase, but in A549, there was no increase at all. So we took these cells, and I wouldn't spend much time on these slides, and we infected them with the virus. And upon qPCR of the cells, what we saw is that in HepG2, we see an overexpression of the viral genes inside the cells. In M08, it was very high. In B12 also, we see overexpression. So whereas in A549, we didn't see that, because in A549, as I have mentioned, though we see overexpression of ACE2, TMPRSS was not expressed. So that's why the diabetes did not have much effect on, or hyperglycemia did not have much effect on the lung condition. With that, we went to the in vivo models. I will quickly go to the in vivo models. That there are 2 mice models which are available, but we had to import them. That's why we use Syrian golden hamster as our in vivo model. And we standardized the infection. Infection model was standardized in hamster. So we could use 10 to the power 4 PFU per ml. We use different concentration of the virus, and we saw that like 10 to the power 4 to 10 to the power 5 PFU per ml is good enough for the viral infection. And the 4 days we used it for multiple days, and we see that up to 4 days, if we keep it, we see the viral infection is setting. And what we see is that we standardize that again with remdesivir. I will quickly go to the next one. That the way we see the infection is that hamsters do not die of SARS-CoV-2 infection. Only the body weight is changed. So the uninfected animals had increasing body weight, whereas when we give when we infect them with the virus, the body weight is reduced. But even upon remdesivir treatment, the body weight is not increased, maybe due to toxicity. That's what we see when we see the lung. When we check the lung, we see that the lung is treated with the infected lung is infected lung is also shrunk. But when we use remdesivir, we see a lot of toxicity there. So we did a PK study with, just 1 minute. I will have just 2 more slides and we'll finish it. So we did a PK study in 4 animals, and when we gave intravenous injection, we saw that the PK was not very good because we didn't have a formulation for this molecule. But we saw that the elimination phase is very long and the distribution phase is only for few hours. So we used the intranasal route also. We used the intranasal route also to administer the drug, and we saw that increased the PK. And but through the intranasal route, we could see little bit of when we did through IV, there is little bit of toxicity in liver, but heart, kidney, spleen, everything was fine. But when we did it with intranasal route, only we see little bit of toxicity in lung, but other organs are fine. So with this, I will stop now, thanking my team of 8 people who have done multiple different things. And if there are any questions, I would like to take that and then complete it.
Thank you, Dr. Das. This was a wonderful presentation. And it's so interesting, but unfortunately, my job as a moderator, I have to push you. I'm sorry for that, but I can allow one quick question from anybody. Yeah, please go ahead.
Hello, ma'am. My question is related to the recombinant ACE2 protein. What I have understood by your presentation, that the recombinant ACE2 protein, it will sequester the SARS-CoV-2 virus, and thereby it will inactivate it. But the ACE2 domain will also, the recombinant protein, is it also there that it will also sequester the angiotensin 2, which binds to the ACE2 receptor?
No. So here, it might, because it's actually an active protein. That's a good question, because this is another thing which we are checking it out, that this is an active protein. It can bind to it can actually sequester the angiotensin 2 peptide. So, but that will only increase the activity of it, which is good for the human being. So it will decrease the hypertension.
Okay, ma'am. And one more thing, what I have understood is that when this recombinant protein will be present over the cell surface, so it will only sequester the virus, but it won't allow entry of the virus inside the cell. Is it so?
Right, because it is neutralizing the virus and it is not allowing the virus to come near the natural receptor. We are not blocking the natural receptor, because the natural receptor should be there. Because as I mentioned, that if I inhibit the natural receptor, then the blood pressure will go up, because that's the normal function of the receptor. So we don't want to block the normal function of the receptor. Giving excess of this ectopic receptor, which will be circulating in the system. So I didn't say blood, because it can be in the nasal mucosa also, that it can be circulating in the human matrix, and it will sequester the virus, not allow it to come to the cell surface at all. Because whatever we see, that all the actions of the virus happens after the cell enters the, after the virus enters the cell.
So this protein will directly be injected into the blood, or it will be expressed inside the body?
Both No. It will be either the route of administration will be either intravenous or we are trying it as a nasal spray.
Okay, ma'am.
Just
Thank
like
you, ma'am.
steroids, it can be done by nasal spray.
Yes. Thank you.
Okay. Yeah. Thank you.
Excellent. Thank you, Dr.
Thank you.
Das.
Thanks.
So I hand over the mic to the repeater.
Thank you, sir. Our next speaker for today is Professor M.C. Subhash Peter. Professor Subhash Peter is a professor in Department of Zoology at University of Kerala. After obtaining master's degree in zoology in 1983, Subhash Peter earned his PhD degree from the University of Kerala for his research work on the endocrine growth physiology in a freshwater fish in 1989. After holding CSIR Research Associateship for a year, he joined the Department of Zoology of FMN College, Kollam as lecturer in 1991. In 1999, he underwent postdoctoral research training in the Redboud University, Nijmegen, the Netherlands, and availed MNT International Fellowship for 3 years. After obtaining postdoc training, he joined the Department of Zoology of the University of Kerala as reader in 2004. He started a fish stress physiology and endocrinology laboratory in the University of Kerala and began his journey to understand the molecular mechanism of growth-promoting hormones and stress hormones in inland fishes and its response to aquaculture-related stress. He became professor in 2009 and served the University of Kerala as Dean of Science, Director of School of Life Science, Professor and Head of the Department of Zoology, and the UGC Coordinator of SAC DRS2 program on fish stress physiology. He has generated research fund from different funding agencies, including UGC, DST, DBT, and Higher Education Department of Government of Kerala. He established Inter-University Center for Evolutionary and Integrative Biology in the University of Kerala that offers MSc program in Integrative Biology since 2014. Currently, he serves as the Honorable Director of this center that employs 17 people, including tenured faculty, postdocs, research fellows, and supporting staff. He mentored 21 PhD theses, 34 MPhil dissertations, and 27 MSc projects on fish growth and stress science. He has delivered 35 invited talks in international and national symposia, published 52 journal papers, and presented 166 papers in international national conferences in India and abroad. He has visited many universities around the globe and presented nearly 35 invited state-of-art lectures in international national conferences, including Global Meet of International Society for Ecological Modeling held in Austria in 2019. Further, he had organized around 15 international national workshops, conferences, symposia related to fish growth and stress science, including CEIP 2015, that led to a GCE publication. He holds visiting scientistship of Yokohama City University School of Medicine, Japan, as elected Fellow of the Royal Society of Biology, London, Member of Society for the Experimental Biology, London, and SEB Ambassador to India. He uplifts students' community. He holds membership in National Academy of Science, India, executive membership in India Society of Comparative Endocrinology, and executive council membership in International Society of Fish Endocrinology, France. As an editor of General and Comparative Endocrinology, a journal of Elsevier, and founder secretary of Society for Evolutionary and Integrative Biology, he manages science of life and explores the innovative and interdisciplinary dimension of fish physiology and endocrinology for creating more employability. This was in brief about Dr. Subhash Peter. Over to you, sir.
Yes. Thank you so much for the introduction. Am I audible? Hello?
Yes, sir. Yes, sir.
Okay. So at the outset, I'm so happy to appreciate the effort of Professor Roy of the Department of Zoology at Goa University and his team for inviting me to give a talk on integrative biology and the process of life. Actually, I'm so happy that it's a pleasure for me to give a felicitation to Professor Roy for his initiating his post-retirement life, and I wish him a happy, fruitful life. So it's my great pleasure to give an outline on why we do integrative biology in the University of Kerala. And I'm so happy that it's all be a part of an effort to understand the process of life in all life forms. And as we see, it starts with a simple thing that when you manage or advocate or promote science, we have a two-way of integrating two things like basic science, natural science as we see, and that opposes the applied biology or medical sciences. So we have an integration with all these kind of things, taking care of different concepts and principles from sciences, and we also take the technological inventions, and so it's all been taken together, and we go ahead with our discovery and invention. And what we see is now the innovative science. So normally when we talk about science, there are a lot of principles on ethics that all come in. But at the same time, we have a way of giving more stress on the innovation. And at the same time, we always look on the integration. So these are the two ways which we tackle science. So science and technology, as you see, stands for the innovation and integration. When it all comes together, when we need to integrate the technology and even the humanities too, to this. And as a result, ethics, together with ethics, we always have an aesthetics. So it is a way of living science. And these two things have to be taken care. So this is a real modern biology where the integration of knowledge is being taken from the different disciplines. And why we do this, to go deeper into the biological systems where we have managed to see a network of process of life processes. And we take all these things to develop ourselves and also to address the basic issues related to the development of biology-based questions. And of course, we need to touch all the societal issues always. And when we do this, there is an individual contribution that all been taken care from our background, and it gives us a new insight. So the concept of integrative biology lies on that it is both approach-oriented and attitude-related one. So when we practice science, we need to integrate all levels of biological organization, no matter whether it is a development biology, physiology, or functional biology, or in the ecology, taking care of the evolutionary questions. We extend those things right from the micro or nano level to the mega level, and even the ecosystem is being taken together. So this is totally diverse perspectives have been addressed here. And when we do this integrative biology, we always touch with the platform with the multidisciplinary approach, and we address all these issues related to this the particular questions. As a result, we utilize a number of diversified techniques and approaches for ourselves and addressing all these things together. So there is a need of taking care of the integrative approach. So integrative biology, of course, is part of the integrative science, and the integration of research and teaching is together taken together and with the arts. So what is the difference between the specialized biology, where we have a reductionism approach, compared to the holistic approach that we employ in integrative biology? So specialized biology, because of the reductionism, we always address a specific question and having a small canvas, maybe in a big magnified canvas. But integrative biology addresses a general question and how related with the multidimensional views. So reintegration of the subdisciplines of biology together with the modern and other things in the engineering computation, so that we get a more in-depth on the quantitative and qualitative processes that are happening, the life and life processes. So the application, of course, it's really a conceptual thing where you have to even look on the sustainability of our science, and also looking on the conservation aspect of the bioresources around us. So there is a complexity that we find in the natural system. So we have to deal with the many organizational scales, and we need to take all this structural organization. For example, when you look on the modern times, we have a habitat destruction, and that would be a lot of impact on the structural organization of the biosphere. And that has to be tackled, and these are the future perspectives. So the importance is that there is a generation of new hypotheses and questions. So as a result, we say that it is the 21st century science, and we need to process this further. At that point, we always see that whether it is a new or an old one. And when you look on the new concept, it is refreshing actually. It is a synthetic approach has been taken and examining the difference. So even, for example, the meta-analysis also been taken, because the result that from the reductionist approach is being taken and makes together, making the modeling system. So it providing many organizing principles so that we can diversely apply it and integrate all the things to use expertise from the different people around us and give more advancement in the science. At the same time, this approach is old, and it gives a sense of history, search of looking back to the unifying principles that are all being finally taking care of the complex answers and questions. So at that time, it is a conceptual entity also. So these are the advantages where we have a more edge over making synthetic hypotheses, techniques, and ideas, and that all definitely promote the science of life. So this is the reason why we are all being taken together. And at our university, we have been looking on the integrative stress physiology that are been there in the vertebrates, particularly in fish and mammals, where we use these models to look on how the integrative science or integrative biology applies. So we have been very fascinated with the way that how animals perceive stresses, and as a result, they respond by stress. So there's a great physiology works on it. And when you look on the stress physiology, we start with the simple thing like home is the best way of looking on both the functionalistic approach as well as the integrative or holistic approach to understand the process of life. So let me explain a little bit of the basics of the stress, how it will happen. And there are a number of definitions or accounts on how the stress happens. This is nothing but the stress is a state of homeostatic disruption, and that all demands some kind of physiological correction. So this is a simple picture where we have a non-stress stress as a result of a stressor of any sort. We or the organism or the life form gets stressed, and when the time permits, it goes back to the normality by post-stress. So lots of physiological processes have been affected and induce the stress response. This is nothing but the physiological reactions to the stressor. Homeostasis is now getting disrupted, and as a result, a disturbed allostatic load is being put together on the homeostasis. And a number of neuroendocrine signals and endocrine stress axis, especially the brain sympathetic chromaffin axis releasing catecholamines, and the HP or HP axis in the fish, where they promote corticosteroids, and they all come together and work on it. And there's a great physiology of the stress. So we find a lot of modifiers of endocrine stress like hypothalamus, pituitary thyroid axis releasing thyroid hormone T4, T3, and even T2. So there are so many other endocrine axes, particularly the estrogens. They're all having an influence on the endocrinology of stress. So this is a cartoon where we have a recovery response after when you put in the stress response, and it happens through the stress acclimation. And recovery response is being now termed as a ease response, And that is nothing but a recovery response that happen in a post-stress acclimation. And we have seen that serotonin and melatonin as a neuromodulators or neurohormones, they have an important role in the ease response. And we are taking another paracrine, endocrine molecule like nitric oxide also been taken, and how does that all involve in the ease and ease response in fish. So we classically look on that corticosteroid and catecholamines having, and along with the thyroid hormones, determine the stress phase of a fish. And we don't have much on knowledge on the ease phase. So that's what the reason we are all looking on the serotonin. So let me look on serotonin. So I will be talking about the three important molecules which are all being taken care, and how do they promote ease response, so that the magnitude of the stress response can be put together and make the animal come to a recovery phase. So as you see, the serotonin is an important neurotransmitter, combined neuromodulators, having a work on different kind of proteins. So for us, we are been very interested in looking on sodium pump. It is nothing but a hydrolyzed enzyme utilizing energy for transporting both sodium and potassium against the gradient that you see here. So it has a component alpha, beta, and gamma, where they have a different isoform subtypes, and which all come together and make a holozyme, and that all be very important for the transport of sodium and across, and also the sodium regulation at the systemic level. So we have a look on all these things, and we have a classic startup with that. We apply the serotonin into the system of the epithelia harvest from the fish, and we found that the activity, the rate of transport is so high with regard to the 10 raised to minus 8. So in vitro with 15 minutes of exposure, and that actually led us to see how it actually happens here. So the serotonin, compared to the immersion stress, the fish is being immersed in the water because we used an air-breathing fish. So it gets stressed when you are not giving air. So that you can see here with the rate of transport of sodium and potassium. And we find that when it is being given serotonin, the immersion one is actually been lower compared to these two things. So this is a kind of a recovery risk. So that means, again, we looked on the different regions of the brain, which all respond differently, generally. And we also find that it has been very, very well recovered here compared to the immersion one here. So this gives more importance that serotonin has a role in this kind of protein, which is actually transporting the sodium and potassium across the membrane. So we looked on the ionocytes of the fish, where we have a chloride cells rich in the gill lamellae. This is the lowest one, is an air-breathing fish, which that shows the ionocytes as seat of sodium pump protein, which is been linked and cross reactivity is been taken together. So when we see about the integration, the fish ionocytes, we have branchial ionocytes, sorry for the overproduction here, the renal ionocytes, and also the intestine. So here a great amount of integration happens between the ionocytes scattered in the different body. So if you are looking only on the liver, brain, the branchial ionocytes, it may have a different response. If you are not looking on the renal or the other part, it is incomplete. So the systemic ion transport has been very important, and we need to have an integrative approach to address the issues. So that is the way that we take a lot of work on this type of integration. And this is what we find it. So it is a control shows where immunoreactivity has also been different distal distal tubules where you have ionocytes. It's all different here. And you find there's a gain of immunoreactivity here in the proximal and the distal tubule by the serotonin. And here also in the intestinal part, it gets recovered. This is another switch where we looked on this, how the phosphorylation, dephosphorylation happens in this protein. And as a result, we find the PKA, PKC led the phosphorylation and its inhibition here in the PKA in the kidney. This is like a differential switching is happening there. Likewise, we have intestinal where all are being switched off. So there is a pattern in the signaling of switching on and switching off these proteins. And even we look the differential regulation of alpha 1, A, B, and C isoform transcript in the fish collected from the different gill, kidney, and intestinal epithelia, and we see that differential regulation. As a result, a kind of higher level of alpha 1 in the gill, it's all been taken. So along with the one switch, there will be a compensation in the other organ epithelia. That all happened. So this is a cartoon where we see that how the HT is being taken in the differential, and it's how they affect, how this all been, the different epithelia is being taken for the regulation of the systemic. So there's a shift in the regulation that can happen here. And another molecule that fascinates us is nitric oxide. As you see, these have a lot of chemistry behind it. And we are all very happy to look on the inducible NOS, which has been very important here, particularly during the stress condition. This table shows you how the agonist and inhibitor kind of how do they increase the NO levels there in the blood or in the gill tissues here as a result. And we see that it is being recovered at least partly here in the LNAM treated, which is nothing but an inhibitor of inos. And we find that that kind of a system works along with the chloride cell distribution in the branchial, where you find this is nothing but the chloride cells or the branchial ionocytes here. And this is the collecting tubule or distal tubule or the collecting tract where ionocytes are being tested. So the differential staining could be seen there. So what we see here is the rate of activity with the gill and kidney of fish treated with both SNB and NAM, where we have a kind of differential regulation that we see here in both are. One is providing the NO here, it is not providing here, and at the same time it is providing here. But so the differential regulation here, so in the gill it is upregulated, but here in the kidney it is downregulated. So that's the reason why when we need to have a specific organ systems to be taken together to give an account on how it actually operates. And this is a recovery process where that you see here. So likewise, we extended our work and we find that it all been very highly regulated, and this is nothing but the inos immunoreactivity here with the imaging, and that's all be there in the recovered fish. It is being inducted. So that means inos is getting activated, and that as a result, you get more NO available in the system to counteract, to promote recovery. This happened in the kidney as well, and I would say that we looked again the protein abundance there in the system, and the activity is being followed by the reactivity, and also the staining is being taken here. This all address that. There is a recovery response where NO is being involved. So likewise, we see here in the kidney also the same response, but it in another opposite way. So this is all very interesting that the response of a particular epithelia, either in the branchial region or the intestinal region, they all have a different response compared to, related to their systemic regulation. So this is very, very, so that is a result of integrative approach. Here also we find there is a kind of even PKCs where the LNAM is being taken, and that has been lost here in the other conditions. So likewise, we have again taken another molecule. It is again a part of the tryptophan metabolism. Serotonin is being produced, it's active during the day, and melatonin is active particularly during the night. So these light dark hormones are responsible for engaging, involved in the recovery responses. So that's the reason why we also looked on the melatonin. So again, we've looked on the non-stressed and the stressed conditions of the epithelia. This is the branchial epithelia where we see the round ionocytes are being well immunostained because of the alpha subunit of intensity pattern, and the protein is also being taken. And when you have a recovery, even in the non-stress, there is a response to the recovery, and that's all been there. And similarly, what you got the distal tubules, where you have this type of integration, especially, but more predominantly proximal tubule ionocytes. The renal ionocytes are better compared to the stressed one. These are the stressed fish renal tubular ionocytes, and they're all having more advantage over this. This has been exemplified here in the protein abundance and also here in the intensity pattern. And the intestine, which is, there is no much more difference because the melatonin is synthesized more there in the intestine. So this is a case of work, and when we looked on the different transcriptal abundance here in the different regions, we also see that there is a differential response. Alpha 1 B is really been active here and here, and but B is very low there in the gill and kidney, and in the intestine it is in the opposite direction. So the differential regulation is also being taken together. So the ease phase is being taken. As a result, we have a cycle of stress ease phase, which always happen in our body, and when the stress comes in, we have a kind of activation of the stress phase, result in the corticosteroid and catecholamines. And when you are putting in the recovery, melatonin, serotonin is being taken together. Of course, cortisol, the data is not been given here. Cortisol is also been very active, and we find a paracrine, autocrine, neurogasotransmitter, NO is also been involved. And we have also gathered the evidence for a T2, a metabolite of thyroid hormone, having an influence on the ease phase, together with E2, estrogen is also been. So these are the hormonal status where they give the entire system of integration happens. So there's a lot of crosstalk between the receptors of these hormonal receptors. They work together, tune with the hormone stress receptors like corticoid receptors or even the adrenal receptors. So there is an interplay of all these receptors, and even the non-receptor, non-genomic response also being taken together. And there's a beautiful way of integration. And this is what happening during the ease response. So, and stress ease cycle is being addressed in that level. And as a result, what we see is an integrative stress physiology is taken as an integrative biology area where we could bring more insight on how these molecules, signaling molecules, can work together and make it more viable, how it actually support life. And that's all. And the message of this one is we need to have our sleep well, and we need to eat well. And of course, as far as the work is concerned, we need to do good work to keep away the stress and be at home. And I acknowledge the support of my present and the past students in the lab in the University of Kerala, funding agencies like DSC, UGC, DBT, and of course, even from the government of Kerala also providing us this kind of a support to work on. And I acknowledge the support from my wife, who is also a partner of our research collaboration, WALSA, for their support. Thank you all. And once again, I thank Professor Roy for giving me this opportunity to interact with all of you. Thank you all.
Thank you, Professor Peter. It was excellent presentation as usual.
Thank you.
Yeah, now the floor is open for question.
Please.
If there is no question from the other participants, I have one question, sir.
Please, sir, please.
Yeah. The thing is, it is fascinating for me, this physiology thing, because I did my physiology, MSc physiology, zoology from Nagpur University under PD Prasad Rao.
Oh,
So
great.
yeah, so I was inclined to this physiology thing from then onwards.
Yeah.
So
Professor
he
Ari
taught
is
us.
also doing the same kind of work there with NO and all the stuff.
Yeah. He has done an enormous work with the fish brain physiology, and particularly the pituitary. And
Yes.
if you know Professor Nishikant Subedar's
Sure,
team,
sure,
yeah.
sure. We
So
are on the team in
am
endocrinology.
I am aware about their work and all these things. It is
Yeah.
always gives me immense thinking that actually how brain integrate with this endocrinological system, so that every time and then we are exposed to the stress, at the same time we are going back to our normal kind of behavior at the same time within. How do you respond to these queries rather?
Yeah, it's a great question. Yeah. So in fact, as a physiology worker, we started from other things, but we now fascinated with the brain. How do they actually integrate this? As you know, the nervous system and the endocrine system, or the physiological processes, they all work together very synchronously. And as you see that even the brain parts, regions, segments in the different regions, they respond differently. They work differently. Even when you have an active mid part, and this compared to the other regions, they are different. So likewise, the neurotransmitters are being released. Once it actually senses the stressors presence, the body utilize the nervous system to communicate fast, and as a result, the entire system is being augmented. And that all been taken from the hypothalamus, and hypothalamus pituitary, the specific organs, endocrine system is being again activated. So these two together work. There is a lot of cross talk between, and it favoring many other even paracrine particles like signals like nitric oxide or carbon monoxide, even ammonia. They all work together and they have a possible integration. And these two nervous system, endocrine system, immune system, they work as the centers of chemical coordination, work very well, and that all been initiated by the neural stimuli. So as a result, we started again with the new brain regions, and we got more fascinating results, which I haven't touched here in this talk. So likewise, and again, an interesting part is whatever you talk about brain of vertebrate, it is incomplete. What you gain from the brain function is incomplete as far as the stress response is concerned, because the gut region, the GIT, is also contributing the brain function. So this brain gut axis is a major task. So once you have a activated stress response, the brain is active. But when you are keeping them in the and filling the tummy with the good food, then the gut region is getting activated, and which has a role in the recovery response or ease response. So the brain shift is being seen in during the recovery phase and the stress phase. So this is beautiful. Did I answer to your question, sir?
Yes, absolutely. It is.
Thank you.
Thank you. There is one question.
Yeah.
So good afternoon. This is Professor
Pragna.
This is Professor Pragna Parikh from the MS University of Baroda, sir. Sir, it was excellent to listen to you. It was very nice. Of yours, sir, it was so, you had made it so easy to understand the stress response and the ease response.
Oh yeah.
So thank you so much to make us understand. Thank you so much, sir.
Yeah, thank you. I really welcome that. That's a process that we have differentiated, because
yes, sir.
It's not been taken, ma'am. Actually, this concept is never been taken that much by the people around us, because we have a group of stress physiologists. They
yes.
No, come on.
Yes, sir.
What
Yes,
said
sir.
is not right and right. Even some of our people are asking me, is it a typographical error or something?
Yes, sir. So that is what. Yes, sir. That is what I was to tell you, that this ease response has now opened new avenues for the scientists to
Yes.
work on that aspect.
Yes,
Thank you so much, sir.
exactly.
Thank
In
you so
fact,
much.
In
Yes,
fact, I have
exactly.
another observation that when somebody talks about stress physiology in fish in Antarctica, so in Antarctica, if you see the temperature, it's already sub-zero. So if you take that fish from there and put it in the normal temperature, that itself is a stress for the fish.
Sure,
While
sure.
while having in sub-zero is not a stress for the fish.
No, no. So every organism is being tuned with the environment, and they are very happy to. They have the evolutionary advantage over living in the challenging condition. And if you impose something else, then it is a great way of stressing. And what we see here, that there is always this kind of a differential players for the ease and stress response. And it's beautiful. It's really, we enjoy looking on this physiology. Thank you so much for your perception and suggestions. Thank you. I really appreciate it. Thank you.
Thank you, sir.
Okay.
Our last invited speaker for today's session is the chairman of today's session, Dr. Anshuman Sarkar. Let me give you all a brief intro about Dr. Anshuman Sarkar. Dr. Sarkar is a professor in Department of Biological Science at Birla Institute of Technology and Science, Pilani. Dr. Sarkar obtained his PhD degree from Department of Biotechnology, University of Pune, India in 2004. He did his postdoctoral training in Cold Spring Harbor Laboratory, New York, USA for 3 years. He joined Department of Molecular Biology and Microbiology in Case Western Reserve University in Cleveland, USA as a senior research fellow. Later on, he was offered associateship in the Cancer Biology Department in Cleveland Clinic Foundation, Cleveland, USA. He joined Department of Biological Sciences, BITS Pilani KK Birla, Goa Campus, Goa in 2009. He was awarded Young Investigator Pilot Project Grant by DBT, Cancer Biology Mission in 2015. He was selected as Fellow from All India Society of Cell Biology in 2016. He has more than 27 publications, 37 conference proceedings, and book chapters published to his credit. He has also organized 4 workshops and conferences. He has handled various projects funded by Government of India and guided 6 students for doctoral degree. He is recognized for his research on anti-malarial drugs potential in treating breast cancer. So this was in brief about Dr. Anshuman Sarkar. So over to you.
Thank you. Is it a full screen now, the slide?
No, it's in slideshow, sir.
Is the slides changing?
No, sir. Can you just go back to your slides and just go to the slideshow tab on the first ribbon. After animation, there is a slideshow. Okay, just go on the ribbon next to the animation. There is a slideshow, sir. No. Just come down.
It's down, right? Slideshow.
Yeah, after animation, no. After on the file, home, design, the slideshow is there. Your slideshow. Up next to animation.
Oh, okay.
Yeah, just click and come to setup slideshow. Setup slideshow. Yeah, click on browse by individual window. Click on that and say okay. And now you can go ahead with the regular PowerPoint show. Yeah, that's it, sir.
Yeah, thank you. Thank you. Yeah, so I on the onset, I know everybody's hungry, but I want to say thanks a lot, Professor Ramavallabh Roy, for giving me this opportunity to present my small work, what I do in the Department of Biological Sciences, Pitspilani Kekebirla Goa campus, with a small group of research. That's what I am going to present today, old wine in new bottle. Why old wine? Because we are repurposing a drug, which is very commonly known drug, that is basically anti-malarial drug, quinine, which we commonly known as. But I have used it to treat the cancer patients, and how it works. We have got very startling results, and some of the results we have already published in few good journals, as well as here I am going to present in today's work about how we are going to use this drug to treat the breast cancer in human. So basically, as there are plenty of students are also attending this symposium, so I would like to remind all of you that you all know that cancer is a group of disease. So where we basically we deal with, it's a group of disease. So we basically we deal with a plethora of diseases like unregulated cell growth, invasion of the cells, spread of the cells from one site of the origin to the other parts of the body. That is called as metastasis. And then it is sometimes it is very difficult to manage the entire issue with the cancer. So there is different type of cancer, like there's a carcinoma, sarcoma, adenocarcinoma, leukemia. All these are different type of cancers. So out of these, we have chosen here the breast cancer, which is most common female cancer. In the almost 25% of the cancers in female are the breast cancer, and there are it's increased to 1 in 8 cases very recently worldwide. According to WHO, in 2018 alone, there is 6.27 lakh death worldwide, which accounts for almost 15% of the cancer death in women. And there the issue is the breast cancer is also rise in India. It is both in rural as well as in the urban side. A recent report in breast cancer statistics showed around 1.62 lakh new cases registered, and 87,000 reported deaths are there already. And 50% of the Indian women suffer from the stage 3 or 4 of breast cancer. So you can imagine how deadly this disease is. This is ICMR data, where 27% of the women diagnosed with cancer have the breast cancer. This is, I am talking about in Indian context only. 1 in 28 women will suffer from the disease, I'm not saying that they are suffering, but in future they will suffer from this disease. And 50 to 60% of women diagnosed with breast cancer in India die within a year. This is very, very shocking, and because there is a lack, and all problem starts with a lack of knowledge. And there are, in the breast cancer also, there are different types. The most most deadliest breast cancer is IDC, or invasive ductal carcinoma, and that is also very recurrent cancer type, which accounts almost 80% of the breast cancer worldwide. It originates from the milk duct. You can see in the figure, it is given a picture of the breast cancer and ductal carcinoma. So it originates from the milk duct. It eventually invades the fibrous tissues of the breast outside the duct, and the entire proliferation takes 1 to 2 months for each division. And in case of cancer, when you talk about a division, it's division in a geometrical progression rate. That means 2 to 4, 4 to 8, 8 to 16, like that way. It's just a huge amount of growth, and immediately around the breast, there are plenty of immunological glands, and you have lymph node. So everything is get affected, and cancer takes the cell metastasize so quickly that it can invade. The moment it goes to your lymph node, immediately it can circulate to other parts of the body. So the problem is mainly with the breast cancer or any type of cancer. The problem is a lack of awareness about the breast cancer, and its early diagnosis is the biggest problem. Therefore, it all depends on the personal or the individual care. And if detected early, breast cancer can be easily treated. And generally, the disease has a very high survival rate, which is a good news. So if we all combine together to fight against this particular cancer, we can save an enormous amount of life in the world. This has been taken from one report recently came out in Current Breast Cancer Reproduction. So here it shows what I am going to talk about, what is invasive carcinoma. If you see on the top panel, if you see the rightmost figure, the panel, which shows the invasive carcinoma, and here are the ductal epithelial cells. So you see that how the entire cell, they rupture the cell barrier, and they invade in the other tissues, and they comes from a low grade DCIS to the invasive ductal carcinoma. So we are dealing with this, the final stage of the invasive carcinoma. We are dealing with a small molecule named as quinacrine, which is a 9-aminoacridine molecule, which is commonly used in malaria treatment. It is also used in giardiasis. It has a weak antiseptic and antibacterial activity, but it is formed when an additional benzene ring is fused with the pyrimidine structure of quinoline nucleus. And quinacrine promotes autophagic vacuoles development through LCB3 and impedance of P62 selective autophagy receptor. You know how the autophagy work inside the cells, and Professor Bandyopadhyay in the morning, he showed some slides on this autophagy model, and it's the same way the autophagy receptor works in case of cancer as well. And quinacrine inhibits DNA damage repair and induces DNA damage, which may be associated with a drug intercalation with the DNA. In addition, quinacrine interaction alters chromatin model. Okay, the entire DNA which comes in the chromatin, so it alters the chromatin structure as well, which is resulting in chromatin trapping. And further, there are reports which prove that quinacrine activates apoptotic signaling through several pathways, such as inducting of P53 and enhancing the receptor binding to the trail, as well as arresting cells in S phase. Very recently, we have published a paper in Oncotarget where we have shown that how quinacrine binds with a protein and how it affects the cell cycle regulation, particularly G1S phase. So in a nutshell, currently there are increasing recognition that need rationally designed drugs that act on several different proteins and pathways altogether. The anti-cancer mechanism of quinacrine is very complex with many potential cellular targets, and it works through the signaling cascade, so signal transduction. And you know, signal transduction is so complicated. If you pull one string from one corner, another string might be pulled from another corner. We never know. We don't even know. And particularly in case of a disease like cancer, there are a couple of pathways are very important, like EGFR, then Notch, then Wnt signaling. So all the signaling cascades, they are all interrelated. And particularly in case of breast cancer, EGFR is very, very important molecule. And inflammation, which Professor Bandyopadhyay also told us today morning, how it is involved in cardiac myocardial infarction, as well as we have seen that in case of cancer also, inflammation is now considered as another hallmark of the cancer biology. In cancer, there are 6 hallmarks are there, basically. One is evasion of apoptosis, another is growth signaling autonomy, then evasion of growth inhibitory signals. The 4th one is inhibition and metastasis. 5th one is unlimited replicative potential. And 6th one is angiogenesis. Along with that, as per the new model, the inflammation is being considered as a 7th hallmark of the cancer. And quinacrine's anti-inflammatory effects would seem to increase its potential utility in anti-cancer drug. And its effects of multiple key signaling pathways is very, very important. And therefore, it gives us immense understanding. And I will show you that where we are in using, is going to use quinacrine as one of the chemotherapeutic reagent in the cancer treatment and conditioning treatment. This work we have already started in Manipal Hospital in Goa with the help of the Goa Cancer Society. I was talking about crosstalk. Crosstalk means how each molecule talk among themselves in a complicated manner. Like when I talk about the crosstalk between NF-kappa B, which is a transcription factor, as well as P53, there are many lines in this crosstalk. And there are different pathways or the signaling cascade where the AKT can be activated by I-kappa B and I-kappa, as well as through the phosphorylation of P53 through P65. Then there are AKT-mediated MDM2 phosphorylation, which also inhibits the P53 stabilization, which requires for each cell to get it stabilized. Otherwise, it will enter into the continuous proliferation of the cell cycle. That is why P53 is also known as a master regulator in the apoptosis. In addition to that, NF-kappa B has been shown to directly regulate the level of MDM2 mRNA and hence as well as its protein as well. So one promising aspect of quinacrine is its simultaneous ability to inhibit AKT, to induce the P53 pathway, and further, which is going to block the NF-kappa B pathway. So that's the whole crosstalk in essence. What we did, we cultured MCF7, which is a breast cancer cell line in the lab, and we have exposed to different concentration of quinacrine, and you can see how the dose response curve is going to affect the quinacrine in 24 as well as 48 hours time, and the reduction of the cell number. This is how the cell morphology getting modulated after exposing to quinacrine. Here you can see the morphological changes very precisely. the formation of this kind of specific protrusion in the cells. And then you can see the spicules have been formed inside the cell around the nucleus. And these are all kind of things that happen once a cancer cell, particularly MCF7 breast cancer cells, are exposed to quinacrine. Here, I'd like to also mention that there are a particular protein which is known as small GTPases, or like RAC, Rho, CDC42. They regulate the cell morphology, and cell morphology is one of the very important phenomena in cancer biology. And if a cell is going to be cancerous, it will come out from its normal morphology and it will change its morphology. It will become more flattened, and it will make all kind of protrusion. It will try to sneak out. There, you know, there is a also known as a phenomena which is known as EMT, or that EMT phenomena, where the cell is very much need to change its morphological modulation to enter into epithelial to mesenchymal transition, or the EMT. This is another data, small piece of data, where we have, this is a very simple experiment where we have used the quinacrine along, we have exposed the cells to quinacrine, and we have used ETBR, that is ethidium bromide, and orange dye. There is a, so what it shows, if you look into this picture, this side is a hook staining, or hooks is going to stain the nucleus, and you can see how the nucleus is getting broken, and ETBR staining along with the orange dye, which is going to that this shows how the apoptosis has been happening in this particular slide. So if you increase the concentration of quinacrine, you can see the cells are dying. You have those yellow color cells, which is apoptotic cells, nuclei, and orange cells, which are already apoptotic cells. Further, if you count those cells, here is the kind of count and how it's going to happen at 24 hours and 48 hours. So again, coming to the stress biology that already Professor Peter had told it, all of us, that how stress is going to respond. And everything, you throw something on a cell, it is going to either generate a stress response, or it is the protein is going to backfire, and there are other proteins which will be going to express inside the cell to negate those stress. But at the same time, there is a production of reactive oxygen species inside the cell, which is very, very essential for a cell to die. So which we have shown that if we expose to quinacrine after 2 hours, 4 hours, and 6 hours, how much is the expression of the reactive oxygen species. This is a typical wound healing assay, which we have done in the lab. We have exposed the MCF7 cells to the quinacrine, and then we have done the wound healing assay, where we have shown the cell, how the cell migrates, how the migration is getting inhibited. And we have shown is the cells, so here you can see the cells started rounding off and detaching from the monolayer, but did not show any significant change in the denounced, this is called as denounced region, denounced zone at 24 hours with the increase in the concentration of quinacrine. The denounced zone at 24 hours up to 20 micron, 20 micromolar is this much. And you can see that like with 15, its maximum doses is 15, how the denounced zone, how the quinacrine is affecting the cell migration. Same way, we do another way to show how the migration capability is getting stalled with the help of quinacrine. And you can, this is called as a matrigel migration assay experiments. You expose the cells to matrigel, and then you stain the cells, and then you count the each cells, that how much cells have been migrated So here, if you see this picture, these are the migrated cells after exposing to quinacrine. You can see the less number of cells are getting migrated with the increased dose of quinacrine. And we have also shown that how, while exposing quinacrine, how the ERK pathway is getting affected. The quantification of phospho-ERK release from the MCF7 exposed to quinacrine has been documented here. You can see that furthermore, a 48 hours exposure to quinacrine resulted in a significant decrease in the level of phospho-ERK from the 0.7 to 5.2 nanogram per liter with cells. And this is cytochrome C release. And these are all different parameters to show that how the cells are entering into the apoptosis, which is a very, very important phenomena to stall the cell. So this is the expression of RAC, Rho, CDC42, whichever I was telling you that why it is so important. It modulates the cell physiology, cell morphology. Everything is modulated through these small GTPases. We have done these expression pattern recognition through the RT-PCR, and you can see the expression pattern, how it is getting changed with the RAC, Rho, and CDC42 in the mRNA level, as well as we have also done with the P53 also. It was taken care how the P53 is regulating after exposing to quinacrine. This is another but very important phenomena is HSP70. HSP70 is a very important protein, which is particularly a chaperone, and that's how it protects the cell, and it makes the cell ready to negate with any kind of stress. But in cancer biology, the HSP70 means there is a change in the HSP70 expression means a lot, because cells are unable to cope up with the stress producing with this quinacrine, and that's how it is entering into the apoptosis. At the protein level, we have shown here both like at the mRNA level as well as the protein level. So you can see how the HSP70 is getting exposed, and at the mRNA level at 24, 48 hours, as well as in the protein level 24 and 48 hours. So finally, in summary, quinacrine induces ROS in ductal carcinoma cells. Quinacrine causes alteration in the cytoskeletal changes through small GTPases, which I was telling you, and reduction in ERK linked with HSP70 modulation that mitigates cancer promotion, and quinacrine affects cancer cell metastasis. A better small molecule to treat IDC. We have also shown, we have generated tumor in mice model, and we have shown that how the tumor is getting reduced in its size after exposing to quinacrine, which is published, which is also got published. And this work has a tremendous importance in case of basic research before we enter into the final league of doing it on the patients, as well as we have already done it on the mice. Now it is the time for us to go to the patient and to the clinical trial directly. The work is done by this girl, Angela, and that paper has come up in the Biomedicine of Pharmacology, which is one of the good journal. And we got support from DBT, Government of India, Goa Cancer Society, and BITS Pilani Goa campus. And thank you. If any question, I'm glad to accept this question. Thanks.
Good afternoon, sir.
Yeah.
I have a small question. Thank you for this lovely talk. Sir, what effect does this quinacrine has on normal cells? I mean, does this has any side effect?
We have seen that it has hardly any side effect on the normal cell. We have already done it.
Okay, sir. Thank you.
Thank you, Dr. Sarkar, for a nice talk.
So there is
Now,
one question in the chat box.
okay. Dr. Sarkar, please see the chat box. There is one question for you.
Yes, I am going to it. Is there a possibility of adenomas in the breast become malignant if there is a continuous recurring? Yes, there is a chance. There is a chance. Yeah. Yeah, there is a chance. Yeah.
Hello, sir.
Yeah.
Sir, I would like to know any toxicity studies you have done?
Yes, we have done with a normal cell line. We have done toxicity study. We have done also in the mice the toxicity study, because you require all those data, otherwise it will not satisfy neither the funding agency nor the journal.
Right. And sir, this dosage that you had given, sir, maybe I have missed the slide or you said, but what was the dosage?
It has started from 5 micromolar till 20 micromolar.
So how did you decide, sir? How did you decide on this dose?
There are already published papers where they have used this.
Okay, sir.
But if you, the moment you go to the mouse model, there is a per kg, again the doses are changes. Okay.
Yes, sir. Yes.
Yeah,
okay. Thank you, sir.
Thank you, sir.
Yeah.
Honorable Chairperson, Mr. Anshuman Sarkar, our most valuable invited guest speakers, delegates, and participants, it is my privilege to offer this vote of thanks. I would like to thank and appreciate our first speaker, Professor Arun Pandyopadhyay, for providing information on dysregulation of cholesterol transport and compromised inflammation in coronary artery disease revealed by plasma proteomics. I should mention deep meaning of appreciation to Professor Prakash Babu for his explanation on effect of rabeprazole treatment, mitochondrial membrane potential in Plumba Gene treated cells for the treatment of brain tumors. I would like to express my sincere thanks to Dr. Ashwini Kumar for giving excellent coverage on One Health approach for human well-being by delivering information on emergence of zoonosis and its impact on human race, impact of human actions on emerging infectious diseases, impact of emerging and re-emerging infectious diseases. I'm also very grateful to Dr. Manjula Das for providing insight of a novel recombinant protein, Solase2, to fight COVID-19 by providing information on protein construction, characterization, and its effect and comorbidities. I would like to express my heartfelt thanks to Professor M.C. Subhash Peter for providing information on integrated biology, its concept, importance of integrative biology, and result of integrative approach on stress. And I would like to acknowledge my gratitude to Dr. Anshuman Sarkar for chairing this session and for exposing us to his research on the potency of anti-malaria drug for treating breast cancer by using quinacrine. I once again thank all the speakers of today's session for sharing your research, information, and views on different aspects. Finally, I would like to take this opportunity to record my heartfelt thanks to all the delegates and participants for patiently listening to all the speakers. Thank you. We will take a lunch break now and resume with the session sharp at 2 p.m. I request all the presenters to be ready after the lunch break.
Thank you.
Thank you, sir.
Thank you, sir.
Dr. Sarkar, okay, the session is not closed. Session will break now. We'll join after the lunch at sharp at 2 o'clock.
Okay, sir.
We'll join at 2:15.
Okay, sir.
We'll join at 2:15. By 2:10 or 2:05, everybody please join. And thank you, Dr. Sarkar, for chairing the session.
Thank you, sir. Thank you.
Nice talk also.
Thank you. Thank you.
So I have an announcement. Since Professor Anshuman Sarkar sir has got some emergency meeting, this session will be chaired by Professor Roy sir on his behalf for all the oral presentation. Yeah, thank you.
Thank you. Thank you.